Welcome to class number 9, beginning of Unit 2, and we're going to be talking today about pathogenic cocci. So we're going to cover 4 different pathogenic cocci. The first one up here is staphylococci, and these occur in clusters. So these are gram-positive cocci that occur in clusters. Now the second one on the list, streptococci, these are gram-positive cocci, but these will generally form chains or pairs, or they can even be in singles. General statement though, we're going to say they form chains. Because of the way they divide, they extend, split down the middle, extend in half, and divide that way. So occur in chains, pairs, singles, but just remember the chain part of that. The last one we're going to talk about here, or the last two, are Neisseria and Moraxella. And these are 2 pathogenic cocci that are diplococci when seen, and they are gram-negative as well. So these are gram-negative diplococci. And one important point about these is they vary quite a bit in shape. So you may get some really large ones or some smaller ones, kind of a sprinkling, almost like a confetti in there. You get a lot of different sizes of them. And they're mashed really closely together oftentimes. So a descriptor that's used by many is hamburger buns. So diplococci that are smashed together kind of have this flat adjacent side. So kind of looks like a gram-negative hamburger. So that point that I made about Neisseria and Moraxella varying in their size, this has to do with, or this is in contrast, I guess, to staphylococci and streptococci, where within the species, they are very uniform. So if you look over an entire slide, they'll all be the same shape, same size. You won't get a lot of variability, where in Neisseria and Moraxella, you will get quite a bit of variability. Now I bring up this slide just because if we're talking about gram-negatives, remember gram-negative being that pink reddish color that we would see in there. And so if we had gram-negative cocci, it would be these pink reddish colors, but that pleomorphic coming up there. So again, we'd get a lot of variability in the size of these. So we'd have gram-negative differing in sizes. Got a little ahead of myself on the last slides, but this is the gram-negative diplococci. Beautiful image here of it. There you go, gram-negative diplococci. And these aren't quite smashed together enough to be hamburger buns in this image, but just use your imagination. So mush them together a little bit and you'd have those flat adjacent sides. Now let's start off with this slide by bringing up this statement here. So pathogenic cocci are the most frequently isolated group of organisms in the clinical lab. The important part of this phrase is group. So the most frequently isolated group. Because if we're going to talk about an individual organism, hands down, E. coli is the most commonly isolated organism in the clinical lab. And we'll talk about that one a lot more in depth later on. But for now, pathogenic cocci are the most frequently isolated group of organisms in the clinical lab. So as a group, we're talking about staphylococci, we're talking about streptococci, we're talking about Neisseria and Moraxella. So both gram-negative and gram-positive. Now, within this group, there are some pretty dangerous organisms there that are capable of causing serious harm or death to the patient. And of these ones that are able to cause a lot of harm, a lot of them are able to produce pus. So the term we use for pus is pyogenic or suppurative. Really, they're the same, can be used the same interchangeably. Pyogenic or suppurative. So these will create pus in the patient. So you'll have a spot where they go in, cause the infection, and you'll get a buildup of red blood cells, you'll get white blood cells, dead and dying tissue, you'll get organism, the organism itself, the cocci that are in there that are dividing, multiplying, dying, and this builds up the pus there. So it's something that will continue. It can really get out of hand. You can get large carbuncles just full of pus. But the pathogenic cocci, many within this group, are capable of doing that. Now, another term, if you're working the bench and you see this come across, the term would be purulent exudate. And you might see that, and that's just a nice syringe or something full of pus. So, fancy term for pus. Now, another important part about pathogenic cocci is they may be part of the normal flora. And they may just act as opportunistic pathogens. So, maybe they're just looking for their chance to get in and cause infection, but your innate immune system, your skin, other barriers that exist there may keep them from being able to invade and cause problems. But they still have the capability of getting in and causing issues that way. Now, I have a reading assignment for you to go through on page 408. So, how your normal flora will flip over to become opportunistic pathogens. It's a really good read. Make sure you do it before the next test. Starting off with Staphylococci, this is in the family Micrococcaceae. And in the family Micrococcaceae are the two genera, Staphylococcus and Micrococcus. Now, mostly what we're going to be covering in this section is the 4 Staphylococcus species. And we'll just touch on Micrococcus here in just a minute, but Staphylococcus aureus, epidermidis, saprophyticus, and hemolyticus. 4 of the really important clinical pathogens that you really need to know to work the bench in microbiology. As promised, this is Micrococcus luteus. So this is under the family Micrococcaceae, and this is a really cool gram stain to look at. And it's got some fairly identifiable features in it. First off, these will be pretty large gram-positive cocci. So large gram-positive cocci, and they also have the unique feature that they form tetrads. So you'll get clusters of 4 of these stuck together. Sometimes you'll see 8, so little pockets of 8, but they form these clusters of 4, and you'll just see those predominantly all over the slide. Micrococcus luteus will also produce a yellow pigment on the SBA. They'll be catalase positive, and so doing a gram stain and making note of the colony pigment is important in differentiating it from another staph species. Next, we're going to talk about some common characteristics that exist with all staphs, all Staphylococcus. And the first one here that I would like you to write down, the first out of 4, is that they are all gram-positive cocci that form clusters. All gram-positive cocci that form clusters. And this cluster, at least the pathogenic ones are. And the clusters are implied in the name there. So staphyle, I think that's how you pronounce it. Staphyle, this is a Greek term for bunch of grapes. And really, like, when you look at many of these on the slide, they do tend to form a grape cluster formation. So a bunch of grapes is really a good descriptor for this. 2 images of Staphylococcus. So we have Staphylococcus aureus on the left here, and Staphylococcus epidermidis on the right. Now, yes, you would report these out as gram-positive cocci clusters because it is important. Clustering is a unique feature. But you can just see how some of these are clustering, and with the aureus, there's quite a few in there, so you're seeing some of the clusters stick together, but if you broke those out, or even if you look over in the top right on Staphylococcus epidermidis, you can see some pretty good clusters there too. Really looks like a bunch of grapes, Concord grapes in this case because they're gram-positive cocci, so some delicious Concord grape clusters that are just waiting to be picked and eaten. The next point, point number 2, of the 4, the common characteristics to all staphs, are that these are medium-sized dome-shaped colonies on an SBA. So they're really pretty uniform. You don't see any that are huge, just take over the colony. That doesn't happen with staph. That would be extremely weird. But you get these medium-sized dome-shaped colonies on the SBA. And as a reference, they are generally smaller than the enteric gram-negative rods. And the pigment of them, though we don't necessarily categorize them based on their pigment, pigment's usually differing quite a bit more in Staphylococcus aureus, but white to light gold is generally what we see. And if we see it going more light gold, it's more often a Staphylococcus aureus, but again, like we talked about before, if there is variation within the species, then you can't call it that. So we don't use pigmentation to identify it, but it might be something that helps you or makes you second guess a test that you saw. So anyway, pigment, white to light gold, and these are uniform, medium-sized dome-shaped colonies. Here is a plate at 24 hours that's been pulled out of the incubator, and you can see some of these smallish, medium-sized colonies on there. Small, I guess, if you look at them in comparison definitely to the gram-negative rods that are on there. So you can see the big difference in size. The gram-negative rods will grow much quicker, divide much more rapidly, and so you'll get a much larger colony than you will with a gram-positive cocci. Now this is another colony, another plate that we have, a little bit bigger colony, but still not anything that we would consider large. We just have about a medium-sized colony that is white, and that is an important part of it. If you see these white colonies, typically you would be leaning towards the Staphylococcus because this is a commonly encountered organism in the clinical laboratory, and that white color is something that you get used to seeing. Now you can see that still in comparison to the gram-negative rod, it is still quite a bit smaller. All right, one last slide just looking at that difference between the two. You can see those good white colonies there versus the gram-negative. Unfortunately, this slide also highlights my cheapness and my willingness to just smear out a plate and re-incubate it to try and get growth for testing the next day instead of resubculturing. So ignore that part of it, but main point here that I just wanted to highlight is, again, I just wanted to hit this one more time, show you one more image, because I do think repetition with seeing gram-positive versus gram-negative is one of the most important things you can do when trying to differentiate these. All Staphylococcus are catalase positive, and this test will really help us differentiate the staph from the strep. So you come in, you're looking at a gram stain under the microscope, and you see that you have a gram-positive cocci. Now your next step should really be to go to a catalase test. If it's catalase positive, you're in the staph area. If it's catalase negative, you'd be looking at streps. So just as I mentioned, and this is the flow chart, we saw it before when we were looking at plating media and talking about the mannitol reactions. But just like I said, gram stain, we have gram-positive cocci right up there near the top. So the next thing in this flow chart is a catalase test. Positive determines what other testing we'll be doing, if it's going to be pushed towards the Micrococcaceae family or mostly Staphylococcus, or if we're going to go the Streptococcaceae route. So you may be asking, what is a catalase test? I'm going to cover that much better in a few minutes, but I just want to show you catalase is just a slide test that we can do. Very simple spot test, a little bit of hydrogen peroxide, look for a bubble reaction, which is a positive catalase reaction. Now the last point here, point number 4, that is a common characteristic to all staphs is that these are common normal flora, found all over the body, all throughout the body. But I do want you to add a note that there are 2 places where they are not found as normal flora. And that's in the vagina and in the small intestines. So it's not in the small intestines due to the bile, the presence of bile in there, that will inhibit Staphylococcus. So take a quick glance at Norma here, and I have little red dots next to all the Staphylococcus spots of normal flora. So the skin, stomach, outer ear, urethra, not in the vagina, not in the small intestines, but is in the large intestine, mouth, oropharynx, nose, and in the eye. So really can be found everywhere else as normal flora. So I said we'd come back to the catalase test, so here it is. Now, catalase test, again, this will differentiate your staphs from your streps. And there's a couple different methods you can do this. Slide method, you could put it into an empty agar plate that has no agar in there, just grit it out, make little spots so that you can smear and drop a little catalase on there. And using a plate is actually a good method to do, because then you can cover up the plate so that you don't aerosolize the bacteria. That is a concern. It's something you should be careful about. But using the slide method, on a glass slide you would place a few drops of 3 to 5% H2O2. H2O2, of course, also being hydrogen peroxide. Then you would take a loop and you'd grab 3 to 4 colonies and you would mix that in and try and smear it around with the hydrogen peroxide. So, if something is catalase positive, you'll get rapid profuse bubbles, and that will indicate that the catalase enzyme is present. Now you have to be really careful. For it to be truly catalase positive, you need rapid profuse bubbles, not weak, wimpy little bubbles showing up. So need to make sure of that. But if you do have an organism that has that catalase enzyme, then when it breaks down that H2O2, it will create hydrogen and oxygen gas, and so you'll get bubbles that come off of it. And again, you got to be careful with this because those bubbles can potentially aerosolize pathogen. So be very cautious, make sure you're covering it. Don't just do this out on the bench top and be hovering over it, sniffing it, type of thing. So an example there, you can see a catalase positive is very strong, big bubbles coming off of it, rapid profuse, and really it would be great to see it in motion. But catalase negative, you can see there's some bubbles in there. It's pretty weak and wimpy though, so that would be a catalase negative. This is more rapid profuse bubbles, so oftentimes it'll just knock off the loop or the stick or whatever you're using to mix it in, and when it does that, if the colonies of the organism are sitting in there, they'll just continue to bubble and bubble until they really break it all down or it's all completely dead and destroyed. A couple of precautions to take note of, and you should already have these in your course packet on page 2-5. Beware of wimpy bubbles, like I just said. Those will be negative. If you're not getting strong, rapid, profuse bubbling, then it's not a true positive. Enterococcus faecalis can cause wimpy bubbles. Another thing that will cause wimpy bubbles is red blood cells. Red blood cells are torn apart with hydrogen peroxide and they'll give you a false positive. Which, side note, hydrogen peroxide is actually useful to get blood out of things because it will really destroy the red blood cell, but you'll get that bubbling that comes out, so you know it's working. But since red blood cells will give a false positive, you want to be careful about not cooling your loop in the agar, because when you do that, you're adding that catalase enzyme. When you destroy those red blood cells, the catalase enzyme is stuck to your loop. So then if you grab some colonies and put it in there, whatever you had on that loop is going to bubble as well. And you also want to be certain that you have isolated colonies. I'll hit on this a number of times, but it is something that's so important to make sure that you don't have something that's mixed in there. This is probably one of the biggest sources of errors in the bacteriology lab is people grabbing the wrong thing, reporting the wrong test, and it just leads you down the wrong path with it. You want to make sure that you have isolated colonies so that it is a true positive or negative. As we go through each of these organisms of Staphylococcus, each of these individual species, we're first going to be talking about laboratory identification. So we're going to go through all the laboratory identification of the individual staphs. And then from there, we're going to move on to some of the pathogenicity and some of those things that they have, the what's in the toolbox type thing. And then some of them will be talking a little bit more about the epidemiology of them. Now, starting off with Staphylococcus aureus, colony morphology on an SBA is medium white to buttery color. So like we looked at before, they'll kind of have a white color. They can be white, which it makes it so that you really can't differentiate it from a Staph epidermidis, but you can get a yellow color, so a pretty good buttery yellow. Not quite as yellow as Micrococcus luteus, but kind of that tinge to it. Buttery is a good descriptor. Now the smell of these, they are described as smelling like dirty gym socks. So like when you walk into a locker room, that smell, which is convenient because that smell of the locker room is most likely caused by Staphylococcus aureus. So just smelly staphs that are on the skin that when just heated up and warmed in moisture and all that mixes together just right, you get that nice stank of a locker room. So dirty gym socks is a good descriptor for the smell. For hemolysis, these can be beta or gamma. So we may have a good clearing of hemolysis around the colony, or we may have nothing. There may be no hemolytic reaction whatsoever. So where we have this variability, and same thing where we have the variability with colony morphology, we cannot use these for ID. So the color and the hemolysis cannot be used for ID because they are not consistent within individual species. So Staphylococcus, you cannot use the beta hemolytic or gamma hemolytic reactions to say whether it's one thing or another. In contrast to Streptococcus, where we have the Streptococcus flowchart right here, where we would use that beta hemolysis, alpha hemolysis, or the gamma hemolysis to be able to determine what test we would then do next. So if we know it's Streptococcus, that's great. The hemolytic reaction matters a lot, but with Staphylococcus, not so much. To highlight that point, here is a Staphylococcus aureus species. Strong beta hemolysis surrounding this colony. Very large zone of beta hemolysis. And this is one we use here in our lab frequently on campus. But then here is another Staphylococcus aureus strain number 2. And this one has absolutely no hemolysis. So gamma hemolytic. And I mentioned that pigment can change as well. Pigment's not uniform necessarily within a species. So this is a Staphylococcus species that kind of has more of a dull white, kind of grayish look to it. Not a bright white like we've seen with the other Staphylococcus species. So hemolysis can't be used for ID. We can use a plate that is very helpful in identification, which is one we talked about earlier, the MSA. Remember, the mannitol salt agar plate. Now, Staphylococcus aureus will be mannitol positive on this plate, though it may take a little bit of time, 48 hours. And so if you had a mannitol negative organism, you'd want to re-incubate that for 48 hours to make sure you get the reaction if it is there. So mannitol positive, very useful in helping you to identify the organism. Very useful, though again not definitive, because Saprophyticus is ruining the really solid result that you were hoping to get, because it can be mannitol negative or mannitol positive. So still helps you quite a bit. If you have a good strong mannitol positive on the plate, it helps you to lean towards that direction. Again, further testing would have to be done to confirm that it is truly a mannitol or a Staphylococcus aureus versus one of the coagulase negative staphs, CNS. Remember that mannitol reaction, we would be looking for the yellow color to appear. So mannitol positive would be a yellowing of the agar. So then what would Staphylococcus aureus look like on a colistin nalidixic acid agar, on the CNA plate? Would it grow? Yes, it would. It's a gram-positive cocci. That's what we use to try and isolate gram-positive cocci is the CNA plate. So on the CNA plate, it would be white. We would get growth, again, which means that that is a gram-positive cocci. So on this plate, we would have 4+, kind of a dull white, but this is a CNA plate, or an SBA. It could be CNA or SBA. If it was the only organism growing, it would be one and the same, grow through all the same. CNA is just there to knock down the gram-negative. So what would be the reaction then on a MAC plate? Sub this out on a MAC plate as well, not sub it out. If we primary plate on a MAC plate, what would we expect to see? No growth. We wouldn't expect it to grow on a MAC plate. Remember what the MAC plate is looking for. We're trying to isolate gram-negative rods, so it's really the opposite of that CNA. So if we have a gram-positive cocci, put it onto a MAC plate, nothing will show up. So Staphylococcus aureus would be the same as epidermidis, as streptococcus, as any of the others. So they won't show up on a MAC plate. No growth at 48 hours. Staphylococcus epidermidis, the colony morphology on an SBA would be a white dome shape. So again, a white dome shape for Staphylococcus epidermidis. And these ones are typically, you can't call it a rule or anything like that because it's not consistent, but oftentimes are lighter in color. So the epidermidis species can often be a pretty good white color and they're most often, almost always, gamma hemolytic. I make my comment on the color of the species, so epidermidis oftentimes more white, but really cannot differentiate based on color. So difficult thing to do. It does maybe clue you into one thing or another, but you will frequently be wrong if you try and just make that assumption too. We already talked about this. So what is the hemolysis of Staphylococcus epidermidis? I just mentioned it. It's almost always what? Almost always gamma hemolytic or non-hemolytic. NH, if you want to shorthand it. So on a mannitol salt agar, we've talked about this one before too. What would the MSA reaction be? So in contrast to Staphylococcus aureus, Staphylococcus epidermidis is man negative. And this will show up mannitol negative at 24 hours and 48 hours. And you really want to make an entry at both time frames to show that you took it for the full 48 to make sure it truly was a negative reaction, not just one that's going to change later. So here we have our mannitol plate, our MSA. So say this is at 24 hours. What are we going to do next? First thing we'd do is record no growth at 24 hours, and the second thing we would do is re-incubate it. So put it back in the incubator, let it go for another 24 hours. If we get any yellow at all, that is a mannitol positive reaction. So any yellow at all. And you can clearly see this is really reverted back the other way. This has gone pretty strongly mannitol negative in the other direction. We've seen this plate before, but it's more relevant now. We have Staphylococcus epidermidis on the left side and we have aureus on the right side. And you can see pretty strongly how strong that acidic reaction is with Staphylococcus aureus. Utilizing the mannitol, and so then it is dropping that pH, the pH indicator reacts, turns the whole thing yellow. It's forcing it into the epidermidis range even because it's so strong. On a CNA, what would be the reaction we would see? What would we see? What's it going to do on a CNA? Staphylococcus epidermidis on a CNA. It would grow, we would have a white colony. That's about it. So we get growth, nothing fancy, it's on there though. So that shows us that this is a gram-positive cocci that is growing on the CNA plate. Now the MAC, what would we see on a MAC plate? Nothing, right? We wouldn't see anything on a MAC plate. So no growth at 24 hours, no growth at 48 hours. It won't grow. It's a Staphylococcus. Gram-positive cocci won't grow on a MAC plate. Next on the list is Staphylococcus saprophyticus. Now, colony morphology on this plate, it can vary back and forth. So 50% of strains, you'll get a somewhat yellowish pigment to them, similar to what you would see with Staphylococcus aureus. So again, that's why this yellow pigment does not help in identification. But 50% of the time, it can be a white pigment. So again, does not help in the identification. Back to the old standard plate of Staphylococcus saprophyticus and epidermidis, so saprophyticus being the one up top. And you can see this is more of a white color that you see with this. Hemolysis for saprophyticus on SBA, it's almost always non-hemolytic. And MSA reaction, what is the MSA reaction if we had saprophyticus growing? Mannitol negative or mannitol positive? Which one would it be? It would be either of those, right? Mannitol negative or mannitol positive. So, can get either reaction out of it. Now, even though we can go either way with this reaction, the MSA reaction is typically a little bit weaker. So, like I was saying before, Staphylococcus aureus can be a very strong mannitol reaction. So it uses it up a lot quicker, but with saprophyticus, it'd be a little bit weaker. So still mannitol positive, but maybe not as strong as Staphylococcus aureus. Some Staphylococcus aureus species can be. Now, next on the list is the CNA plate. So CNA, this would just be exact same as what we would see on the SBA. So we can see either a yellow pigment or a white pigment to them, because they will grow on it. Now the next plate, MAC plate, again, maybe you're shouting it at your monitor right now. What would happen on a MAC plate? Nothing would. Nothing would happen. No growth at 24, no growth at 48. So we're kind of leapfrogging. We finished looking at the laboratory identification of the Staphylococcus. Now, we already went over the catalase when we were talking about the staph reactions to the catalase test. So again, put these, you could even smear the colony on the plate or the slide before you put the hydrogen peroxide on. But either way, hydrogen peroxide mixes with organism. You either get bubbles or you don't. Rapid profuse bubbles is a positive. No bubbles or very wimpy weak bubbles would be a negative. And so we already went over recording the results of that as well, but down below, if we're recording lab results, now let's jump down to that portion right below the catalase test. When we're looking at the plates, we want to document, and documentation is really key when you're working in a clinical microbiology lab. I've said this before, that oftentimes you're not always going to follow yourself on the bench. It's great when you do because you know everything to expect the next day. It's wonderful. But if you're following behind somebody else, you just hope that the person before you documents well enough to know everything that's going on so that you can just pick up and see everything they did and you know why they did it. So documentation really is key. And we do it a little bit differently than some of the other places I've worked. But in our lab on campus, we'll document it based on the plate. And so up top, we'll write down all the plates we set up. And if this was a laboratory information system that you can enter all these plates, you just add all the plates that were put in under. And usually, if you work in a big enough laboratory, this is all just done with primary plating. So whoever's setting it up, if it comes in as a urine culture, then you have a couple different plates that are automatically thrown in there when a urine culture is ordered. So that the tech the next day can then come in and see that SBA, MAC, CNA, and MSA, for example, were set up on this. So the list of primary plating media that was given. Now, in our laboratory, we would come in, we'd look at the SBA, and we would list it down there. And then from there, we would start going through the different reactions, and we'd take a look at the plate that we have to see what organisms are growing. So starting out with our SBA, we would pick it up. We'd be analyzing all the different quadrants, and this is a plate that has a lot of growth on it. So from those individual quadrants, we have quad 1, 2, 3, 4. And we can see just by looking at it that we have at least 2 different organisms on here. And by the looks of it, colony 1 looks like it may be a gram-positive cocci. Colony 2 may be a gram-negative rod. Can't necessarily confirm that. We would want to do a gram stain, colony gram stain of these just to confirm. But anyway, that's our initial take on this. So in recording these, putting these under the SBA plate, colony type 1, we could write out that this is 4+ white because it is growing out in that 4th quadrant really well. Colony 2, 4+ large gray mucoid. And large gray mucoid is just a generic term that we can kind of use. So you can adjust this a little bit, but I use this as a phrase that on-campus students pick up so that we can categorize things a little bit better. But large gray mucoid often leads us to a gram-negative rod ID. Back over to where we're going to record these results. So that next day, we've documented SBA, 1+ or 4+ white. Colony number 1, 4+ white. And we want to leave a fair amount of space between these two, at least if you're documenting this on paper as we do in our laboratory. So if you're putting this on paper, you need to have room for the different subculture plates and testing and everything else we're going to do to try and identify. Colony number 2, 4+ large gray mucoid. Now we're looking at that 4+ white, and this is looking like a gram-positive cocci. Still should colony gram stain it to confirm, make sure that we know what it is before we're going to report anything out. But at that point, we could say, let's do a catalase test. So we add some hydrogen peroxide to the slide and grab a few colonies, and we mix it in, and rapid profuse bubbles. It starts bubbling like crazy. So then let's go back over to our report form. And we are now going to document that this was catalase positive. Rapid profuse bubbles means catalase positive. So in doing the catalase test, we've now ruled out that this is Streptococcus ace. So it's not a Streptococcus. We don't have to worry about that portion. We now are looking in the category of Micrococcus, Micrococcus ace. Not Micrococcus, I apologize for that. Micrococcus ace. So the family, that family, and then we're going to start breaking it down. So we got to start looking at what comes next. Now that we know it's in that family, we will want to do a coagulase test. Coagulase is the next step in this flow chart. The coagulase test is the definitive test for Staphylococcus aureus. So this can be used for identification, positive identification on a positive result. And that's because Staphylococcus aureus is the only staph that will produce the coagulase enzyme. So next step in that flow chart, if we have a positive coagulase test, we can call it out as Staphylococcus aureus. If it's negative, it goes into the coag negative staphs, and so we would have to do further testing on it. Now, I say it's a definitive test. There may be some staphs that are positive. There are some others out there. I'd say there may be, but there are other staphs out there that can be coagulase positive. But the only human pathogenic staph that is coag pos is Staphylococcus aureus. The others are animal pathogens, and we can just disregard those. Unless you're doing horse testing, in which case pull out the micro textbook and look up further testing to confirm that it is a Staphylococcus aureus or another type of staph. The coagulase test uses rabbit plasma. And in doing this, the rabbit plasma will have fibrinogen in it. So then if our organism has coagulase, if it's capable of producing coagulase, it will cause the fibrinogen to form a fibrin clot. So a visible clot will show up in the form of a clump, or there's other methods that we can use for this, and we'll go over that in just a moment. So there are 3 different methods that we will use to identify Staphylococcus aureus or other coag-negative staphs. And really 2 of these are done in conjunction, but 2 of them are spot tests. The rapid slide method is a spot test. The tube method really needs to sit overnight, at least 4 hours. The rapid latex test is another spot test. And the rapid latex test really has taken over in most clinical labs as the method of choice, just because of the simplicity of it and its definitive results. So very common. We'll talk about that more in a minute though. Rapid slide method. This detects the cell-bound coagulase. Now there's 2 types of coagulase, or it can be found in 2 places really. One place is bound to the outside of the cell. So on the outside of the cell, we have this coagulase enzyme that exists just sitting there. So if this were to come into contact with the fibrinogen, it then would cause it to convert over to the fibrin clot. Now, Staphylococcus aureus is also capable of pumping it out. So creating extracellular coagulase. So it's outside the cell, it's not attached in any way, it's just out there. So this free extracellular coagulase would then float into solution. The rapid slide method works somewhat. It's produced by some strains of Staphylococcus aureus. But the tricky part here is that we could call it positive based on this, but we really can't call it a true negative. So if you have it, since it's only produced by some strains of Staphylococcus aureus, we haven't fully tested if it is Staphylococcus aureus. So if it's negative with this test, we're not done yet. If it's a positive, then yes, we could report it out as Staph aureus. But the tube method, and this is where I was saying these are joined together, is because if you have a negative rapid slide, you would then need to set up a tube method anyway, because all strains of Staphylococcus aureus would be positive in this, because they all put out that extracellular coagulase. So the rapid slide method, we are trying to detect that cell-bound coagulase to be able to get good clumps in there. So what we're going to do is mix rabbit plasma with 3 to 4 colonies on the slide. Again, I already mentioned this, but here's the part I want you to write down. If it is spot coag positive, that is definitive for Staphylococcus aureus, and you will see clumps of fibrin. You'll see visible clumps, and you won't be able to mix them up. You won't be able to get them to break up. Now, if it is spot coag negative, you must do the tube method, because, again, we can have some that are rapid slide negative, but then tube positive. And there are these Staphylococcus aureus species that are a little bit slower to react. So it takes them a little bit of time to make and excrete that free extracellular coagulase. So 4 hours to overnight for this test. And what we're going to do with this one is we're going to mix about 1/2 ml of rabbit plasma with 3 to 4 colonies, put it in a tube, and we'll incubate this overnight at 37 degrees Celsius, because we want that to be as close as we can get to body temperature to try and mimic that environment that they like. The rapid slide method, so this will work on some strains of Staphylococcus aureus, and this is great because it's quick if we need a quick identification. be able to get that diagnosis for the patient as quick as possible so they can get targeted treatment. So rapid slide, some strains, but if we don't get it, that doesn't mean it's a negative for Staph aureus. We would then need to do tube method because all strains will show up positive with the tube method. And again, just for emphasis, all diagnostic tests must be done on isolated colonies. Do not think you're touching the edge of a colony that, just because it's the left side and it's only partially touching another colony on the right side, they can get through. If you do that, I promise it'll make nightmares for you. So make sure you do all diagnostic tests on isolated colonies. The third one that we're going to go over is the rapid latex test kit. Now, these rapid test kits are wonderful. Beautiful invention. And what they are is they are these tiny little latex beads. Tiny, tiny latex beads that are coated with two things, fibrinogen and IgG. Now, fibrinogen we've already talked about, and that would be your cell-bound coagulase. So the concept of this is that if that cell-bound coagulase is there, the fibrinogen would then attach. You'd get the clot all the same, kind of like we've done with the spot test for the coagulase enzyme. Excuse me. But we incorporate into this as well IgG. So there's this IgG. And Staphylococcus aureus is coated with a protein called protein A. And no other staphs have it. No other Staphylococcus species has it. So this being there on top of that fibrinogen makes this definitive. So if we have coagulase or protein A, this IgG on there, either one would be capable of causing a reaction and making it all clump together. The fibrinogen is an important part of the rapid latex test kit, but really this IgG and that protein A, it's the important part because this should make it a definitive test. The coagulase enzyme with the fibrinogen really helps to enhance it all, but this protein A is the main thing that will cause the clumping. And so we have the stem part, the arm part, the Fab, and the FC, but the antigen binding sites up here will be looking for that specific protein A. So if it finds the protein A, then it will link up to it. So a better way to look at it, a better visual, is we have latex beads with that specific antibody on there. And so the IgG antibodies are targeted towards protein A. Protein A is those little lumps on these organisms which are Staphylococcus aureus, even though they're horribly shaped to be Staphylococcus aureus. Let's use our imagination on this one. But these are Staphylococcus aureus organisms, and they have that protein A antigen on there. So these IgG antibodies are going to start attaching to it. And when they do that, we get particle agglutination. This particle agglutination can be visualized then. And it is a beautiful thing. Beautiful. So what you'll do is you'll add a drop of your Staphylocide latex kit or whatever it is, your rapid latex kit test kit that you're using. So you add a drop of that, and then you will take your suspected colony that you are hoping to be Staphylococcus aureus. When you mix it in, you will get that agglutination reaction and everything will start sticking together. Remember, these are micro beads, tiny little beads that are in there. So we have this uniform blue color, that negative control on the right. And sometimes people can get tripped up, but to have a true positive, you really need to see somewhat of a clearing in the background too. So you get this clumping, and that should be very visual. And then you get a clearing because the beads are all sticking together. So the liquid that's left in there doesn't have the beads floating all around. So this is a really nice positive for Staphylococcus aureus, and this is what you would hope to see if you were testing it to see if it was Staph aureus. So quick comment that I want you to draw what we just went over with the particle agglutination and the drawing of IgG, because it is an important concept, especially when it comes to this test kit, exactly what's happening. So say this test came up negative, Staphylococcus negative. we would want to document that in the report underneath that 4+ white. So back to our recording lab results. So now we have a catalase positive, staphylococcus negative. So they're in parentheses, just kind of as a note to ourselves. We are thinking this is coag negative staph. Or on the flip side, say, let's go back to that reporting thing. So if we did a coagulase negative, if we had a coagulase negative, tested it, no clumping, nothing at all, we would then want to report that. So we would then put that it was spot coag negative. But remember, hand in hand, that's not definitive if we did that. So spot coag neg, we would then want to set up an overnight coag. If we came in the next day or after 4 hours or so, it shows up positive, we would then be thinking it's Staphylococcus aureus. That is reportable at that point. We've made it to identification. You don't need to add that additional date on there because it's implied. It was an overnight test, so the results will just be added onto it. So then we would get down to our organisms isolated, and we would want to report this out to the physician. This is 4+ Staphylococcus aureus and 4+ GNR. We could initially report it out as a GNR, but we would want to further identify it. We just won't take it to that point right now since we have not talked about it at all, but that will be in coming lectures. Now on the flip side of that, say that we were to come in the next day and it was overnight coag negative. So still negative. Then we would be going down. We've ruled out the possibility of Micrococcus by doing a gram stain and looking at the colony. It doesn't look like it's Micrococcus. It lacks that tetrad formation, the large gram-positive cocci in tetrads. So we're leaning towards a coagulase negative Staphylococcus. Now, many laboratories will even just stop at coag negative Staphylococcus because they don't find it very important. Saprophyticus is a very important organism that exists in there, and so other times this may be necessary to go on to do novobiocin susceptibility. And depending on where you work, the protocols in place, you may just look to rule out Saprophyticus and then everything else is coagulase negative staph. But we do want to look for this, and we do in our laboratory here at Weber State. The way that we will do novobiocin susceptibility is sub a plate out. So you want to get an SBA, sheep blood agar. You'll streak out the organism onto the sheep blood agar. And then up in the top of quadrant 1, we're going to add an antimicrobial disc. This antimicrobial disc has been impregnated with novobiocin, so you can get varied concentrations of it. And you'll put it onto the plate, diffuses out into the agar, and if the organism is inhibited by the novobiocin, you will get a zone of inhibition. So you'll get this clearing around where the organism wasn't able to grow because it's been destroyed by the novobiocin. So we can see clearly outside of the zone of inhibition there is colony growing perfectly well, but there is a thick area around that disc where it hasn't been able to grow at all. The way that it's set up in my laboratory is on a pie plate because I am cost effective. And so the cost effective way we do it is to take a plate, mark it up on the back of the plate to segment one SBA into a test for 6 other people to be able to add on to, or for 6 different organisms if you have a lot that you suspect of being coag negative staph. Now, we'll set up controls on this plate. So we'll have a resistant one and a susceptible one. And then we'll set up 6 different patient samples. There we go. There's the animations. So, important part of this is we do not record it as positive or negative. It's not positive or negative. It's either resistant to it, resistant to the novobiocin, or it is susceptible to it. So for that reason, we write an R with a circle or S with a circle, or Novo RES, or SUS for susceptible. So the novobiocin susceptibility, we set that up, and what we're looking for is Staphylococcus epidermidis to be susceptible or Saprophyticus to be resistant. So for that same reason, we set those up as our controls on the plate. This is the back side of the pie plate, which we can see the controls on the right hand side, and they wrote the controls in as positive and negative, but they also wrote in the R and S for resistant and susceptible. So just right there, susceptible, resistant. So epidermidis is susceptible and Saprophyticus is resistant. So in looking at this plate, we have our resistant control up in the top left, and you can see there is no real zone of inhibition around there. Compare that to the susceptible control, where we have a very thick zone of inhibition. Now, bottom left, what do you think of that one? I think somebody screwed up, if you want my opinion. And it looks like somebody put on a gram-negative rod. So they got tripped up early on. They didn't gram stain. Again, always colony gram stain if there's any question as to what it is going to be. Do a colony gram stain, you'll save yourself corrective reports later on. So, this would have saved this person. They ended up writing down that this was a Saprophyticus because it was resistant, right? So they were already off track. So at this point that they were doing novobiocin, they were either going to call this susceptible or resistant and then report it out. So they had no check in place. Make sure to always watch yourself. If something starts looking off, always question it. Question everything with it. So then go back, do a colony gram stain. It's always the test that you can lean back on. Now if you would flip back to the laboratory identification of Staphylococcus epidermidis, I would like to add the novobiocin result on there. And so we want to say that novobiocin, it is susceptible. Staph epidermidis is susceptible to novobiocin. So you can write sus there or an S with a circle around it. And then moving on down to Saprophyticus. Saprophyticus novobiocin result would be resistant, or R with a circle around it. Back to our recording of the laboratory results, we then have novo susceptible. So I know, sorry, I flipped this up a little bit on you. I had a positive result in there, but if we go down spot coag-neg, overnight coag-neg, thinking coag-negative staph, we then have a novobiocin susceptible, and so we're thinking Staphylococcus epidermidis. So that sums up the identification of the Staphylococcus species. So I want to emphasize that this chart is so important for you to memorize. Gram stain reaction, every step of the way, these are very commonly, the most commonly encountered group of organisms. Staphylococcus not alone by itself, but it's part of a large group. We do see Staphylococcus a lot. So you really need to memorize how to identify these because you're going to go over it again and again, and my tests are going to ask you questions on it. The board of certification tests are going to ask you about it. Your future employer is going to ask you about it, or your current employer. And so make sure you have it down, each of the steps of the testing, and just practice makes perfect with it. So get used to going through this flow chart. All right, that concludes class number 9. Pathogenic cocci, specifically Staphylococcus, and the laboratory ID of Staphylococcus. This is class number 10, and we're going to switch gears a little bit. In the last class, we spent a lot of time. It was a pretty long lecture covering the laboratory identification of Staph aureus, inside and out. And in this class, we're going to focus on the various infections that are caused by the Staph species, and in particular, infections that are caused by Staphylococcus aureus. We're going to start off by talking about infections that are caused by Staphylococcus aureus. Now, in the textbook, if you'd like to read along with that, it's 539 to 543. in your textbook. Now, Staphylococcus aureus has a variety of tools. It can be very, very virulent, and it depends somewhat on the strains, but also the opportunities that it's given. Some of the infections that we can see with this, skin infections, wound infections, we can see it disseminating from one infection, existing, just staying in certain spots, waiting to create another infection, and toxin-mediated diseases. It really has a variety of different things it can do. And it exists all over the place. So Staphylococcus are living and reproducing on almost every square inch of your skin right now. They're tolerant to desiccation, radiation, and heat, can survive 30 minutes at 60 degrees Celsius. So they're very tolerant and can exist in some very extremes. So it makes them an organism that we really have to watch out for, because they do have all of these mechanisms to be able to deal with anything that we throw at them, really. Almost anything. The first one we're going to go over is skin infections, or abscesses, and there's 3 different kinds that we're going to talk about underneath this. Boils, and carbuncles, and impetigo. Now, boils and furuncles are the same thing. They both begin as folliculitis. So folliculitis is an infection of the hair follicle, gets in around the hair follicle and has the environment it wants with the nutrients that it wants, so it's able to grow and replicate very easy in there. Here's a really good image of folliculitis taken from one of the textbooks I have here on campus. And it shows in here, you can see, I like that you can see the raised area up around the hair follicle where it's coming out of the skin, because you do get this bump, right? It can look like a zit. And so we have down here Staphylococci. If you look down bottom left, and you can see that it's multiplying, dividing. You'll get phagocytes that are coming in to try and go after the Staphylococci to try and be able to contain it, and it causes more inflammation, irritation. And then we also have that core of pus right there. So the pus is starting to develop around there. And do you remember the term that we use for pus formation? Pyogenic, suppurative. So yeah, we have this pus formation there that's just continuing to build and causing some pain. So creating pressure, heat, redness type thing, causing pain there. So here is another image of a furuncle, and I look at this thing and I want to lance it, just stab it, and I don't know, feel like I would relieve the pain of this person. Not that I'm into that. That's not my thing. I'm not one of those zip popper YouTube watchers. But man, just want to help this person out, get rid of it. I imagine they did after this photo was taken. But we could gram stain this. It'd just be loaded with PMNs or white blood cells and gram-positive cocci all over the place. You'd just see a ton of it. A carbuncle is where the infection from the boil spreads into the surrounding tissues. A good way to remember it is all of your uncles get into a car and have a family reunion on your skin. So, car full of your uncles, a carbuncle. Now, I want you to go look in the textbook for the definition of a sty. Here is an image of a carbuncle. So you can see surrounding tissue all getting involved. It's spread further and further. So this is just a furuncle that got out of control and carbuncled. Big carbuncle. So it's extending deeper into subcutaneous tissues even on this one. The next infection that we're going to talk about with Staphylococcus aureus is impetigo. And impetigo is primarily an infection of children. And the reason for this is the source is generally from the nose. It's common normal flora. Staphylococcus aureus is all over as normal flora in the nose. And so everybody's seen that runny nose kid. My little girl's that way right now. Just, I don't know, she hates to have her nose wiped, and we want to do it all the time because it's just constantly running. So the runny nose kid running around, they make it even worse if they are in daycare or if they're in school. and that runny nose is just going all over the place, and other kids. It spreads very easily among kids. Now there's two different types of impetigo: bullous and pustule. This is an image of a young child with bullous impetigo. And bullous impetigo is described as blister-like. And you can really see why here. So the outer layer of skin just separates away. It's almost like a PPD injection gone bad, but really separates away from the skin. And within that, it's just a blister full of Staphylococcus aureus, just full of it. And really, other people describe it as looking like a bad sunburn, and you can see why. Just those nasty blisters, reddening on the skin. So this is very stereotypical for bullous impetigo. What goes up has to come down. So eventually these blisters will pop or scab over. They'll come down, scab over. And when they do that, you get these crusty scabs that appear all over the skin. Here is a picture of a young girl that these around the nose looks pretty good and crusty, but it looks like further out some of these blisters are fairly fresh. They've just popped, and so they're starting to move into that crusting stage. So they'll start to crust over. Now, I mentioned that bullous impetigo is caused by Staphylococcus aureus. Now, mostly. It is, most of the time, 80% of the time, caused by Staphylococcus aureus. 20% of the time, it's going to be caused by Streptococcus pyogenes. So, bullous impetigo, 80% of the time, is caused by Staph aureus. 20% of the time, it will be Streptococcus pyogenes. This is a picture of pustule impetigo. And man, this one's hard to look at. It looks like he, I don't know. Did you get the definition of sty earlier? Not that this necessarily falls into it, but man, looks like there's a zit right on his eye. But that is kind of what this one looks like. So pustule impetigo looks more like zits rather than the big blisters that we saw with bullous. And they'll crust over and you'll get these black scabs on them. Now this one is caused most of the time by Streptococcus pyogenes. So I don't have percentages to go with this one, but most of the time this will be Streptococcus pyogenes. So compare those two. Bullous impetigo, most of the time is Staph aureus. Pustule impetigo, most of the time is Streptococcus pyogenes. The second common way that we have Staphylococcus aureus causing infections is by wounds. And really from any site here, but if you think about it, Staphylococcus aureus is normal flora on the skin. So anytime we open up that skin, anytime by trauma, scrape, surgery, etc., any of these ways, it's very opportunistic. So if it gets its chance to get in and cause problems, it likely will. The third way that Staphylococcus aureus can cause an infection is by dissemination from a local infection. So for example, a carbuncle or a furuncle or a carbuncle furuncle or impetigo, and it's able to spread out and gets into the bloodstream. It could then cause further problems that way. So right now I would like to define septicemia. Septicemia is bacteria in the blood multiplying, causing symptoms. So they're there in the blood, they get into the bloodstream, they're able to multiply and really thrive in this environment, which is then showing symptoms. You're starting to show symptoms of them being there. Now bacteremia, on the other hand, is the presence of bacteria in the blood, but there's no symptoms accompanying that. So these are often thought of like a transient bacteria. So they got in there somehow, either like brushing teeth and your gums bleed, and the bacteria are able to get in temporarily, but they're cleared out before any of the symptoms start. That's just one example. So bacteremia, presence of bacteria, but not showing symptoms. Septicemia would be bacteria in the blood, and these are causing symptoms. Now, it gets into the bloodstream and it can move around, get to different sites. So one of those places that it can go is to the bone. And it can get into the bone marrow and cause complications there. Basically, it's introduced, moves into bone marrow, and it's a safe place for it to be. If it can find a spot and settle in, it can just sit there. And really, it can go dormant too. So you think you got rid of it, the infection's all cleared up, and it just sits there waiting for its opportunity. So something may happen, may knock down the immune system of that person, or maybe they have some trauma that happens again, kind of knocks it, gets things moving. It can start to flare up again. And so osteomyelitis is one way, and that's an inflammation of the bone marrow. Now, pneumonia can be caused quite a bit by Staphylococcus aureus. And so this is a inflammation of the lungs. This is an infection in the lungs. And when we think of pneumonia, oftentimes we think of the young and old. And that is because they are, again, have less developed immune system, so they're more susceptible to this thing. But they get fluid in the lungs, starts to build up, and I don't know, roughly 10% of pneumonia cases are caused by Staphylococcus aureus. So pneumonia is just one other example. Now the next one down there, endocarditis, and this is more prevalent among drug users. But endocarditis is where the inner lining of the heart muscle is getting destroyed, and in this case by Staphylococcus aureus. So it'll destroy this inner lining of the heart muscle, and it can also destroy the heart valves, which is really bad. You lose the heart valves and you have a lot of problems. Meningitis, most people, when they think of meningitis, they think of viral meningitis, or if they're thinking of bacterial meningitis, they're thinking of Neisseria meningitidis, which we'll talk about later. But meningitis can be caused by Staphylococcus aureus. It is capable of causing it and can get in there, and it can be deadly in that way. This is a picture of my brother-in-law, and man, when I saw this thing flared up, I just had to snap a bunch of pictures of this, and this was years ago. But anyway, he has a recurring infection in his elbow that shows up, and I don't know that he's ever had it cultured. He's never really told me much about that, but they do believe it to be a Staphylococcus infection that is just there in his elbow, and you can see a scar on the upper portion of his elbow. He skateboards a lot, and so he's had a lot of injuries to this, and he's had a lot of opportunity for Staphylococcus to get in there and to settle into his bone. So this is an example of an osteomyelitis. It's just sitting in there dormant, and it's really waiting for his immune system to be compromised and to drop, or it's waiting for something to jar it. That's usually how it flares up, is he'll be skateboarding again, and then he'll land on his elbow, and then he'll end up in the ER the next day because it just takes off. I got a couple more photos. Here is another one, and I really like the lines they draw on there. So you go in when it's initially flaring up, and they'll draw this line on, and they'll say, we have to react if it gets beyond this. We'll need to do something serious. And then you go in again, they're like, all right, if it goes beyond this one now, let's draw another line. So this is kind of the moving goalpost with it, but they did eventually treat it. They usually get it to go away pretty quickly, but it does end up in an ER visit to him because it's really something you can't mess around with. The fourth one we're going to talk about is toxin-mediated diseases, and this really has three main diseases that are caused by the various toxins produced by Staphylococcus aureus. Those will be toxic shock syndrome, food poisoning, and staphylococcal scalded skin syndrome. The first one that we're going to talk about is toxic shock syndrome. And this was first described in 1978. And it was noticed in menstruating young females. So this was previously undescribed disease. But it caught the attention of epidemiologists because there were a lot of women that were getting very sick and some were dying from this. In the end, they linked it to superabsorbent tampons. So this condition was linked to the fibers in these superabsorbent tampons, and the Staphylococcus aureus were able to grow on the fibers and just kick out this toxin like crazy. So those that were using the superabsorbent tampons and leaving them in longer, so overnight, sleeping with them, it was able to grow up Staphylococcus aureus a lot easier. The toxins were kicked out, and this is what was making them very sick or killing them. Now, the specific toxin in this case was TSST1, Toxic Shock Syndrome Toxin 1. And this would get absorbed into the bloodstream. When it would get absorbed into the bloodstream, the toxin would trigger fever, hypotension, shock, and, like I said, in some cases, death. In 1980, the number of cases of toxic shock syndrome was almost 1,200 cases. So these superabsorbent tampons were withdrawn from the market. And you can see what started to happen as they decreased. Now, in 1982, the FDA requires labeling on the tampons to make people aware of toxic shock. And it continues to just decline year after year until you see in 2008 we have probably less than 100, less than 50 even, cases of toxic shock syndrome. So it still exists and still can be a problem, but greatly reduced by getting rid of those superabsorbent tampons and making the public aware of the problem with this. So making people aware so that they're not endangering themselves with those. The next one we're going to talk about is food poisoning. And I bet there's a fair amount of you out there that have had food poisoning. And if you've had it, I've had it a couple times. It's awful. You just completely sick, drops you, horrible. Fate worse than death type thing. And the cause of this is really food that's left out. So Staphylococcus can grow pretty well in mayo-based products. So like thinking potato salad, macaroni salad, all of these nice summer picnic meals that we put out. So you know you've been there a number of times. You've been to a cookout barbecue and the potato salad is just left out. And hopefully those people are putting it on ice or something like that to help reduce the warming effect that will happen in the middle of summer. But if not, if it's just left out in the warmth in the middle of summer, 2 hours or longer, you'll start to get Staphylococcus growing. It allows it to grow because the temperature becomes right for it. Now, what it's going to be doing is it's going to be creating enterotoxins. So it's these enterotoxins that make you sick, enterotoxins A and D. Now, the enterotoxins A and D aren't attached or anything like that, so they're not increased by the growing organisms, but they put out these toxins, and these toxins are preformed. So it doesn't actually, it's not actually caused by the replication of the organism. It's more these toxins that are being kicked out into the food. And so you ingest it, and symptoms start about 2 to 8 hours later. So after ingestion of that, 2 to 8 hours later, you'll that's actually appropriate. You'll start to show symptoms. But those symptoms will be nausea, vomiting, severe cramps, and diarrhea. So the sharding actually fits in there. But yeah, you're throwing up everywhere, just sick. The cramps have you curled up in a ball on the floor. And it will go away a little bit quicker, and if compared to an infection. So I contrast that with salmonella. Salmonella is another food that you can get, or another infection that you can get by ingesting food that hasn't been properly handled. So if you were to get salmonella, that's more like 24 to 48 hours later. So in contrast to 2 to 8 hours, that's how you know that it is a food poisoning issue more so, because it's the toxins. And those toxins get in and they're able to bypass, they're not destroyed by intestinal juices or your gastric juices. So they make it through and they'll just make you horribly sick because of that. Now, the source of the last toxin-mediated disease we're going to talk about is staphylococcal scalded skin syndrome. Say that 5 times fast. Staphylococcal. I can't even say it 1 time fast. Staphylococcal scalded skin syndrome. Now, this is an exfoliative toxin, and it's also called Ritter's disease. But this exfoliative toxin gets in the skin and it causes the epidermis screws me up. Epidermis to separate from the cutaneous layer. Now, it's most prevalent in children. Occasionally, you'll see it in adults, but just like what we saw with impetigo, more common in children. So this will last about 2 to 4 days and has a spontaneous recovery with it. Of course, I have some images to accompany this, and the reason they call it scalded skin syndrome is because it does look like you just took the baby and dipped it in scalding water and pulled it out. So you just get the skin sloughing off, looks burnt underneath, and so fits perfectly in with this image. And you can see baby super unhappy, and it looks like the skin all over the body is just sloughing off. Have another image, this one of an infant, and you can tell the umbilical there is still pretty fresh, but you can just see how bad it is. Like, that leg is sloughing completely off. The skin's going to disappear and it's going to look just as red as right above his penis. It's horrible looking. Would not want to deal with this, but this is one more thing that can be caused by Staphylococcus. One last image. This is a little bit older, so this is a young boy, but you can see again on his face and neck, skin just sloughing off all over. What is it that makes Staphylococcus aureus capable of causing all of these diseases? And really, it's the virulence factors that it contains, these invasive mechanisms. So I have here a partial listing of them, and this is from the textbook. Now look at the Staphylococcus aureus, different virulence factors that it has compared to that of epidermidis. So we have protein A, coagulase, catalase, hyaluronidase, staphylokinase, lipase, beta-lactamase. And it's positive for all these. While epidermidis has some of them, like lipase or catalase, it is missing a lot of these other virulence factors that contribute to Staphylococcus being so virulent. The first one in this list that we have talked about already as the coagulase test is coagulase. And again, this takes fibrinogen, and if the coagulase enzyme is present in that organism, it will turn this into a fibrin clot. So a visible clot will be seen. Now, this is also called clumping factor. And you can think of the benefit of this. So this would trigger some blood clotting to happen. And by doing this, it allows it to make an abscess. So really what the organism's able to do is wall itself off so that the body's immune response cannot come in and, eat up the bacteria that are sitting in there. So it creates this wall around itself that it can just stay and hide. So it's a survival mechanism for it, essentially. And I really like this image from the textbook, page 415. And this shows the bacteria doing exactly that. With that coagulase enzyme, it's forming a clot around itself, so it can just hide out inside there. The next on the list is hyaluronidase. Now, hyaluronic acid is the major component that makes up the matrix between cells. Kind of binds things together. It's the glue that binds things together. So there's very high concentrations of it in connective tissue. It's a thick, viscous substance. Now, hyaluronidase allows it to break this down and break it apart. And so if it's able to break this down, it's able to spread more easily through the tissues. This is called the spreading factor because it allows it to spread more easily through tissues. So hyaluronidase, also a very important virulence factor that Staphylococcus aureus has. Again, from the textbook, this is showing that hyaluronic acid that exists between the cells. So it's not necessarily showing that in that first picture on the left there. But what it is showing is that it's able to divide those cells and break it up. By using hyaluronidase, it's able to get in between those cells and get into deeper tissues. The next one we're going to talk about is protein A, and this is a cell wall protein that is unique to Staphylococcus aureus. A cell wall protein that's unique to Staphylococcus aureus. And we've already talked about this one a little bit. Now, this is an important invasive mechanism because this prevents antibody-mediated phagocytosis. And it does this through manipulating the IgG molecule. So let's take a look at that. This is an image out of your textbook that shows several different ways that antibodies can help clear out bacteria or toxins, viruses, things like that. And it goes through this. There's the neutralization, there's opsonization, which is eating it, and there is agglutination or aggregation. Now, to know how this works, we need to, again, look back at the individual IgG molecule. So this is that same image that we've looked at before. Now you have that Fab portion, the arm portion, and this is what has the antigen binding site. So this is the area that is specific to protein A. Now protein A has this unique characteristic that it can bind not just to the Fab portion, but also to the Fc portion, so the lower portion. So protein A can bind to either the Fab or the Fc portion. So what this is doing is that Fc portion will not be exposed. It's being bound by the protein A. And that's important because if the Fc portion's not exposed, then it can't be attached to the phagocyte. So it won't attach that Fc receptor protein on the phagocyte. If that doesn't happen, then there is no phagocytosis. If there is no bound antibodies, they don't know to engulf it, they don't know to eat it. So it makes them essentially invisible out there to the phagocyte because the phagocyte is relying on that protein A being attached to that Fab portion. It then recognizes it by attaching to that Fc, and then it can engulf it and eat it. I love this picture out of the textbook, and so I use it, but this is phagocytosis happening. This is a yeast cell, so it doesn't necessarily apply, but the Staphylococcus aureus is really avoiding this dire fate. And I love the picture because the yeast cell looks so miserable. I can see a face on there, and it just looks like it's groaning and reflecting on all the poor choices it's made in its life that ended up in it being eaten by a phagocyte. So, yes, Staphylococcus was able to avoid this fate. Now, the cell-bound protein A that's just all over the Staphylococcus aureus is only part of the story with this. There is also 30% of protein A that is excreted by the cell. So it's just kicking out all of this protein A. Now, this is getting out into surrounding tissues or bloodstream, but what this is effectively doing is soaking up all of the IgGs that are being kicked out to eliminate the infection. So it's really this big diversion that's making it invisible still. An example that I like to think of is, like, name any fighter jet scene in, I don't know, whatever movie you like. I can't even think of any right now. Top Gun comes to mind, but they're flying around and somebody gets a lock on with a missile and they shoot this missile straight at it. Now think of that as IgG coming right at it. So the plane that's in front that's just been targeted can kick out flares, just drop the flares all over. And this missile is then directed towards those flares and will blow up or will miss the target. That's kind of like Staphylococcus aureus kicking out all of this protein A. So it's essentially its flare deflecting away, and the IgG then is targeting all of the protein A instead of being able to attach and kill off the Staphylococcus species. So all of that it's able to kick out, it results in a significant decrease in the IgG that are specific for Staphylococcus aureus. So it really gets rid of those that are hunting it. Next on the list is lipase. Lipase is very beneficial, and as we've talked about, Staphylococcus species like to live on the skin and really in many sites on the body, but on the skin in particular, there is oils. So lipase allows Staphylococcus to be able to break down the oils. And it can do this, and it acts as a food source for them. So they break down the oils produced in the sebaceous glands, and it helps them to survive and thrive. Now, all Staphylococcus species produce this. So it's a benefit to all staph species. And epidermidis is one that comes to mind when you're thinking about those that live on the skin. This is a really important one because it's all over the skin. It's normal flora there. So it needs to be able to break down those fats, and it has this lipase enzyme, so it's able to do so. Staphylokinase acts as the antagonist to coagulase in some ways. It's just another way that staph can be awesome at being terrible, is the best way I can put it. But what staphylokinase does is it dissolves the fibrin strands. So remember coagulase, we create this wall. So they're scared that the body defense is going to come in, eat them up, kill them, do whatever they're going to do, tear them up and rip them apart. So they wall themselves off with the coagulase. Now they're sitting in there in this wall, kind of like this little fort they've created, and things may be happening. They want out. They feel safe now to leave the fort that they've just created. And they're running out of space and nutrients because they're still dividing, multiplying. And so they've used up everything they have in there, and they want out now. They want to get out and be able to go cause infections or create their own fort somewhere else. So they can create the staphylokinase, which will then dissolve those fibrin strands. It breaks the clot apart, and then they're able to re-release themselves. So this is an awesome part of it, because there's other organisms that can wall themselves off but aren't good at releasing themselves. So it walls itself off, then when it needs out, it can break that wall apart, go back out, and infect again. The last on the list of invasive mechanisms is beta-lactamase, which also goes by penicillinase. They're the same thing. Now, it's an enzyme that's produced that will break down penicillin. And this is now present in 90% of Staphylococcus aureus strains. So that doesn't mean that there's nothing else that we can do about this, but it does mean that we have to go to other drugs instead of using penicillin ones. So further generations down that are more resistant to penicillinase or that are not affected by penicillinase. But it is something that's developed more and more, and we will see it continue with Staphylococcus aureus. There's a couple things I'd like you to read regarding MRSA, methicillin-resistant Staphylococcus aureus. And the first one is in the course packet, page 2-40 to 2-41. And the next one is a clinical case study out of the textbook, page 543, which is a really interesting case, a fatal case of methicillin-resistant Staph aureus. So we've already talked about a few of the toxins here. The first one, though, is enterotoxin A through E. So Staphylococcus aureus is capable of producing more than just A and D like we talked about before with food poisoning. But these enterotoxins are heat stable, can last in temperatures of up to 100 degrees Celsius for about 30 minutes, so very stable. And entero means the intestinal tract, essentially. So these are toxins that will affect the lining of the GI tract. So again, we talked about A and D and what it will do, but there's 5 total enterotoxins that we have to worry about. Exfoliative toxin, we've already talked about this one. This is the one that causes staphylococcal scalded skin syndrome and also is involved in bullous impetigo. Now, it also is called epidermolytic toxin for really the effect that it has. So, epidermolytic toxin, it's an exfoliative toxin. And the last one we talked about, that's TSST1, so toxic shock syndrome, toxin 1. And this is the one that is associated with superabsorbent tampons, and this TSST1 gets absorbed in the bloodstream. Hypotension, shock, death can occur with it. Now, the third on the list of pathogenic mechanisms of Staph aureus are the cytolytic toxins. And these toxins may be possessed by some, but not all. Some may have all of them. It's just a varying degree of them. But we have these alpha, beta, delta, and gamma toxins. And these are not the same as the hemolytic reactions that we would see on a plate. These are a separate designation. Now, what these are referring to is the individual toxins that are put out that are then toxic for many of the cells in the body. This includes leukocytes, macrophages, erythrocytes, and platelets. And it'll really go in and just destroy these cells. And so it just triggers all sorts of lysozymes to be going off, and it gets released in the tissue. It's like a grenade going off inside your tissues. So these, not quite like a grenade, that's a little dramatic, but it will just destroy any of these cells, just completely lyse them, pop them, and the cytolytic toxins work pretty effectively. But again, they aren't all within every single Staph aureus species, subspecies. They are there in varying degrees. So some may have all, some may have none, some may have a couple, but it is something that can be possessed by Staphylococcus aureus. We've now covered Staphylococcus aureus, if we refer back to this list of the ones we're going to cover. So now I would like to cover epidermidis, saprophyticus, hemolyticus, and actually I'm going to throw in one more to the mix there, and that's lugdunensis. Staphylococcus epidermidis is the most common aerobic bacteria on the skin, and really derived its name from epidermis. It's commonly isolated from the epidermis, so epidermidis makes a lot of sense. And this causes 70 to 80% of the coag-negative Staphylococcus infections that we see. Now, typically it's very low in virulence. We don't see it causing infections all on its own. This is an opportunistic pathogen, so it's waiting for its opportunity to get into a cut, for immune system to be down and to get into some sort of injury or during surgery or something like that. It really has to have a pretty good opportunity to get in there to cause infection. So it's really just waiting for it to happen. It will cause nosocomial infections. Now, nosocomial infections are hospital-acquired infections, or HAIs. And the reason that it's fairly common in these hospital-acquired infections is it can attach to prosthetic devices pretty well. So plastic prosthetic devices. These are catheters and IVs. Now it does this by creating a slime layer, also known as adherence factor. And so it gets in and creates this film within the plastic prosthetic devices and can survive pretty well in there and just shed bacteria out. So Staphylococcus epidermidis is most known in the hospital for hospital-acquired infections, for prosthetic devices that have been left in too long, and we know that it could develop this pretty easily. Now, going along with biofilms, slime layers, and such, I'd like you to go read Associations and Biofilms at page 167 to 169 in your textbook. Staphylococcus saprophyticus, its main claim to fame is causing UTIs in sexually active women and older males. So what it'll do is it adheres to the epithelial cells lining the urogenital tract, and by doing that, it will cause cystitis. Cystitis is an inflammation of the bladder. So saprophyticus, mainly known in this area that it will cause UTIs. The next one that I want you to know about is Haemolyticus. And really all I want you to know about Haemolyticus is that it is normal flora and it's occasionally seen in clinical samples. And it's a staph species, but that's all I really want you to know about it. And the last one, and I'd just like you to jot this one down, maybe off to the right-hand side, but Staphylococcus lugdunensis. This is normal flora on the skin, and it's capable of causing osteomyelitis and septicemia. But its claim to fame is causing aggressive endocarditis. So most known for causing aggressive endocarditis, that's Staphylococcus lugdunensis. Just have a quick note here, and that is that all three of these can be reported out as coagulase negative staph unless they're found in urine or in sterile sites. And in sterile sites, we're saying especially prosthetic devices. Now, if we had it in urine, that would indicate saprophyticus, and especially if it was a young, sexually active female. But whenever we see a coagulase negative staph in urine, we would want to then further identify or at least rule out saprophyticus, and then you might report it out as coagulase negative staph, not saprophyticus, just so that the practitioner knows. Now, sterile sites, you would want to take it further for sure, because especially if it's prosthetic devices, you might be looking at something like epidermidis, or other sterile sites, we might be thinking lugdunensis even, and it could be a cause, it could be an aggressive endocarditis type situation. So these are ones that you would want to work up further. Now, in my lab on campus, everybody always works it up unless instructed to sign it out as coagulase negative staph. But if you're out there working in clinics, it more so, it's a waste of time essentially to work up some of these others that are inconsequential to the patient's health. So that is why it would just go to coag negative staph unless it's in sterile sites or in urine. That rounds out the rest of the Staphylococci group, and so yeah, finishes that up. The next that we will talk about is Streptococci. All right, welcome back to Intro to Streptococcus. This is class number 11, and we're going to talk about some of the general characteristics of the genus, along with the classification scheme of the streps, so which is the Lancefield classification scheme. So we'll talk more about that. Then we're going to cover the first organism on our list of streps, which is Streptococcus pyogenes. So Streptococci are general term that we are using. They are in the family Streptococcaceae, and that includes the genera Streptococcus and Enterococcus. Now, Enterococcus, there's a couple different species that we'll study under them. Really, the main two species that you need to know, period, is Faecalis and Faecium. Faecalis, Faecium, Enterococcus, any guesses as to where these are normal flora? If you guess the intestines, you would be correct. In the intestinal tract. Faecium, spot in the intestinal tract, Faecalis, feces. You know, kind of blends in there. Streptococcus pyogenes, I just mentioned. We're going to talk about that one first. Agalactiae and Agalactiae, pneumoniae, that's my pronunciations for it. It's a number of scientists will pronounce it Agalactiae or pneumoniae. You also have those that'll say Agalactiae, pneumoniae. You get varying things in different textbooks. It's just kind of the region and the scientists you work with based on how you will hear it pronounced. And so, sometimes I even jump back and forth on them because I sometimes decide I like one better than the other. But Agalactiae, pneumoniae, there's the viridin streps, and this is a larger group that includes some that aren't necessarily as severe a pathogen. Some of them can be pretty serious, but mostly we just see them as normal flora. And talked about Enterococcus and group D strep, so we'll cover all of these, but generally referred to as the streps. So this is their common name, so people say it looks like it's a strep if they're looking at it on a plate. Now, we've talked about it quite a bit, so what does the gram stain look like of a strep, of Streptococcus? It would be a gram-positive cocci. And we talked about before how chaining, maybe, the way they divide, common characteristics to see chains or pairs or diplococci, but they can be seen in singles too, which wouldn't be abnormal, especially if we're including Enterococcus in there as well. So how would you report this out if you were to see this on a slide? You'd be thinking, there's quite a few in there, right? High power field on this one, so we have 4+, and this would be gram-positive cocci in chains. There's plenty of chains. This is Streptococcus pyogenes. We wouldn't identify it just based solely on the chaining, but it is a unique characteristic to Strep pyogenes, and these are really good chains that we're seeing here. So here is another stain of Streptococcus pyogenes, and one thing I want to point out in this, beyond the chaining and the gram-positive cocci, which yes it is doing, which then yes would make us think it's Strep pyogenes, but beyond that, down in the bottom middle of the screen, what is that? It's a white blood cell, PMN. So we'll refer to this as a PMN when we see it in there, and we'll want to comment on PMNs that we see. So polymorphonuclear cells, polymorphonuclear leukocytes. So these are usually segmented. We're oftentimes seeing the neutrophils that are moving into the area, so that's what we'll comment on, and that's what they look like in a stain. A little bit different than you're used to if you're looking at a right stain most of the time. This is Streptococcus pneumoniae. Now take a look at this slide, and what are you seeing the majority of? A lot of pairs, right? A lot of gram-positive cocci that are in pairs. And these things are kind of, they get a tapered end to them. So a lot of the streps have the ability to do this, and they'll kind of look elongated at times. They can be kind of this oval, roundish. That is normal for all the streps. But this one, we have lots of cells there, lots of gram-positive diplococci. So lots of pairs, and diplococci is one of those terms that we will use for strep pneumoniae. It commonly will take this formation, so we want to report it because that is important to the physician. In this slide, this is our gram stain control that we use, and we have E. coli and Enterococcus faecalis. So Enterococcus faecalis would be the gram-positive cocci that are in there, the nice, big, round, gram-positive cocci. So on to number 3, but first, just back to number 2, the cellular morphology. We would see just a little bit of everything on this. So we can see chains, we can see diplococci, we can see ones that are just kind of a variation of all things. But chaining is kind of the unique characteristic that we will see in this. But yeah, number 3, colony morphology. So on a sheep blood auger, they will be non-pigmented, and they even can have this translucent look to them. Some people describe it as a ground glass, kind of this refractive somewhat look to it, but they can be a variety of dull as well, and usually a little bit smaller in size. Of course, I say that they're usually a little bit smaller in size, and then I have a staph species that is uncharacteristically small compared to it. Not the best comparison of it, but they will be a little bit smaller. And what I want you to focus on in this image is that it does kind of have that translucent look to it. So in this situation, it would be a translucent beta that we're seeing. So that's colony number 2 up there. Colony number 1 would be that 3+ white. So it's a little more prevalent, and we are seeing a white pigment to it. The next point about colony morphology on an SBA is the hemolysis. Now we can get beta, we can get alpha, we can get gamma. And we can use this for identification. Now that remember that Staph aureus, some strains were beta, some were not, so we couldn't use it for ID. It's not the case with streps. We can use it for identification because Streptococcus pyogenes will be beta, Strep pneumoniae will be alpha. We know these things, and we know that they're consistent within their species. Here's the flowchart that we're going to use for Streptococcus, and again, you will have to memorize this one. So right there at the top, we're talking about colony morphology and hemolysis. So we know that it is a gram-positive cocci, and we know that it's catalase negative. So now we're in the Streptococci realm. Now that we know that, we're looking at the hemolysis on the plate of those colonies. If it's beta hemolytic, it's going to be moved into one of a few different species. Streptococcus pyogenes, if it's strongly beta hemolytic, and if it is positive for some other tests we're going to do, or susceptible. Streptococcus agalactiae, if it runs some of the other tests. Anyway, we'll talk about those ones later, but the main point here, beta hemolytic, if it's alpha hemolytic, then we move it into either viridans or Strep pneumoniae. Non-hemolytic, we're looking more so at Enterococcus or group D streps. So on this plate, we have a Streptococcus pyogenes species, and you can see pretty good beta hemolysis right there around that colony. So even though this one's a little bit fuzzy, you can still clearly see it. And we'd call this one 3+ translucent beta. And remember, we would record the beta on the SBA and CNA. The SBA is the main one that you want to take this from, but the CNA just to correlate it so that we know that this beta is that beta. So that's our way of keeping it straight. Now Streptococcus pneumoniae, these would be really small. They don't grow as fast as even Strep pyogenes or Agalactiae, but definitely not as quick either as the staphs or the gram-negative rods will. Strep pneumoniae is a bit slower, and when we see this, it will be 3+ tiny alpha at 24 hours. So you'll see just these little colonies with some greening around them. There's another way that we use, another method that we use to categorize these, and this is by serological groupings. And in particular with these, we call these the Lancefield groups. And we'll talk about that more in just a moment, but number 5 here is normal flora. So we're going to need to bring up normal flora norma to be able to look over all the spots that you would expect to find Streptococcus. So to norma, if you look around all these different spots, all these different normal flora sites, you can see that they are just about everywhere. They're in the stomach, they're on the skin, urethra, vagina, large intestine, small intestine, mouth or oropharynx, nose, and in the conjunctiva. The only place that we don't see them as normal flora here is the outer ear. And I can't really tell you exactly why, because common sense you would think that they would exist in there. But really found just about anywhere though. So returning now to the Lancefield classification scheme, I want you to turn over to 2-15 in your course packet. And that is where we have some information to fill out about that Lancefield classification scheme. Now, this was developed by Rebecca Lancefield. And if you know much about the history of science, it's actually pretty rare to find many women who made significant contributions. And a lot of it is just because it was the old boys club type of mentality. So really interesting that we have this this person who made such significant contributions. And I won't expect you to know a lot about this. This is mostly FYI, but there's some interesting information if you look it up. I have a link here, whonamedit.com, that you can go find some more information about this, this classification that she developed. The work that Rebecca Lancefield did was based on the components that are found in the cell wall. So what she was able to do is she was able to extract these components out of the strep species. And she was able to then use those on a rabbit, so test those on a rabbit and look for the different serological reactions. And she did find that she would get varied serological reactions based on the components that were in the cell wall that she was able to pull out. Now, based on those, we divide the streps into 3 categories: the C carbohydrates, the lipoteichoic acid, and the non-Lancefield. So let's take a look now at the C carbohydrates. So what Rebecca Lancefield did is she would take these, I like to think of them as antigens, so these carbohydrates, these C carbohydrates, and she would inject them into the rabbit, and she would get these different serological reactions based on which one she was injecting. And she determined 5 groups that had the C carbohydrates, and that was A, B, C, F, and G. Now, group A was tied to Streptococcus pyogenes, what we know to be Streptococcus pyogenes, and group B, Streptococcus agalactiae. Now, there's other ones too. So there's group C, group F, group G, but not D or E, and we'll talk about those more in just a moment. So going down the flowchart that we have, I have arrows pointing to group A and group B, and we do use these terms quite frequently, still used interchangeably. So you may hear somebody say group A strep or Streptococcus pyogenes. They're the same thing. Same with group B and agalactiae. So you can use either term, it's fine. Doesn't matter to me, but just make sure that if you're going to write it out as the group, you need to say group A Streptococcus, not just group A, because we need to identify that it is a strep species at least. The next group that we'll categorize by is the lipoteichoic acid ones. And so these ones have no C carbohydrate in them. And so since they have no C carbohydrate at all, we're looking for a serological reaction that's based on that lipoteichoic acid instead. What this lipoteichoic acid is it's a cell membrane protein that's in there. And we have these other lipoteichoic acids, or teichoic acids, that exist within the peptidoglycan layer. But you can see the lipoteichoic acid, this goes through all the way through the gram-positive cell wall there, and it will embed into the phospholipid bilayer. So lipids in the phospholipid bilayer, lipoteichoic acid. Makes sense to just jam those words together. But this is what we're going to use for that specific reaction. So the grouping that falls under the lipoteichoic acid are the group D streps. And I have under there Streptococcus bovis and Streptococcus equinus, and both of those, I didn't list them in the earlier list of all the Streptococcus species that we're going to cover, but they exist, and you can see by the names bovis and equinus, they're typically pathogens of animals, but they do sometimes cross over, so we do want to be aware of them. But most of the time, we're just going to categorize those collectively as group D streps and not worry anymore about them. Now, the next two on the list there, Faecalis, Enterococcus faecalis, and Faecium, these ones we do worry a little bit more about. So we're going to take these ones, and even though they fall under the group D streps and they are considered a group D strep, most of the time we're going to try and parse that out because we don't just want to leave that at group D strep. Enterococcus, Faecalis, and Faecium are significant enough when found in the body as a pathogen that we want to make sure that we identify it so that it can be treated if it is there. If we go back and look at our flow chart, we have the Lancefield categories down at the bottom. So Enterococcus and group D, you can see, doesn't really matter the hemolytic reaction. If you're not getting them to fall out somewhere else in group A, group B, then typically we'll end up down there near down there where Enterococcus and group D are. Now, we will stop at that point. And like I said, we want to separate those out and take Enterococcus out of the group D. But clinically, in a laboratory, if you're going to be reporting out, group D is fine. You don't need to go any further. There's probably Equinus bovis and a number of others that would fall under that. But we don't have to go any further. That's all the physician would need to know unless they ask for a further workup on it. But group D is sufficient. One other thing that I would like to highlight on this chart is that if we're looking at hemolysis, mainly if we're looking at hemolysis up top, so we already have a gram-positive cocci that's catalase negative. So now the hemolysis comes in, and if it's non-hemolytic, there is a good chance that it is going to be Enterococcus or group D strep. So we know that it could go down that path because we know it's not group A, group B, or Streptococcus pneumoniae. So based on that, we would then be leaning towards Enterococcus group D, and we would still have to do the testing to confirm that, but it's just a good indicator. Now, the third and last group for the Lancefield classification scheme is the ones that are non-Lancefield. So these have neither the C carbohydrates or lipoteichoic acid serotypes. Without these, they really didn't fall necessarily into any category, but there's two of concern here, and that is Streptococcus pneumoniae and viridin streps. So these are two alpha hemolytic streps that will show up, and we'll take a look at the flowchart again. So down the middle here, I've circled in green the pneumoniae and the viridins, and circled in green because they are alpha hemolytic, or a greening of the agar. So these two, it's a pretty good indicator. Once again, alpha hemolytic, if we have alpha hemolysis on the plate, catalase negative, gram-positive cocci, then we are thinking generally one of these two things. So strep pneumoniae or viridins. Here's a list of what we just talked about. So again, it's going to have the major ones on here, at least up top. So we have Pyogenes, group A, group B, Agalactiae, and group C we really don't encounter a ton. It doesn't come up that often, so I'm really not going to cover those a lot. But Streptococcus mutans and pneumoniae, and mutants would fall under the viridins group. And so that's one of the major pathogens in there. Mostly its claim to fame is dental caries. It rarely will cause bacteremia, but can be found in there. So of the viridins group, that's one of note. Now the last one there, group D, Lancefield, again, Enterococcus faecalis is under there, and Faecium. But I'll say that again, we don't typically call them group D streps. We'll usually just refer to them as Enterococcus. Okay, so now we're moving on to the laboratory identification of Streptococcus pyogenes. And this is just a list that I've compiled here of the methods that we will use to get to identification, or the different ways we could get to identification. And we're going to go through each one of these ones individually. Now let's flip back to the flowchart again, and we will look at this ad nauseum. You might get tired of this, but you should get used to it because you need to memorize this thing as well. So we're looking at the colony morphology, and we're looking at the hemolysis. So we see beta hemolytic. Now the next two tests right there, the PYR and bazotracin, and we'll do those, but first we want to look again at that beta hemolysis and explain a little better what is going on there. Now, the lysis that we are going to be looking for, this beta hemolysis, like I've said before, this is like a blow a hole through the plate type of hemolysis. It's just really strong, and you should see a good, strong clearing, a complete clearing around the individual colonies. Now, this complete lysis around the individual colonies is due to a couple different enzymes that exist in there, and that's streptolysin S and O. S is the oxygen stable version of it, and I remember S for stable, oxygen stable, and O is the oxygen labile form of it. So we have these two different streptolysins, and these enzymes work in combination to clear out the red blood cells. So that is why you get such a strong hemolysis in there. So since we have these two different enzymes, the streptolysin S and the streptolysin O, and the streptolysin S is stable. So it's okay in the presence of oxygen. It can be out. It won't break it down. It won't cause any problems with it. But that streptolysin O is oxygen labile, so it does not tolerate oxygen. It will break down and it won't function properly. So the way that we test for the functionality of the streptolysin O to see if it actually is there, if it exists, is we will stab the agar. So we'll get some of it onto our loop and we'll stab it down in. And in doing this, it introduces it down all the way, and you can see in this image here, it shows it going all the way to the bottom of the plate. So you stab it all the way through. Now, I have a little image here. I like to go in 45-degree angle. 45-degree angle doesn't create quite the opening, and it puts like a little bit of an overlap on it so that it doesn't dry out and start cracking the agar as easily. But in any event, the same thing will happen. So you introduce the streptolysin O, and you should see a much better clearing than it just sitting on the surface, because it allows it to get into the agar and it works much more effectively when it's inside the agar. So we'll look at some images here in just a second. Actually, we'll look at quite a few images. So now the first one I have here, you can see you have a pretty good strong clearing. So you have beta hemolysis there. Now, these are STAB O positive. So streptolysin O, we call it a STAB. So a STAB O positive. And these are really good strong positives for it. This is what we would expect to see, is a really strong clearing around where we've stabbed it into the agar. Now, there's another thing on this one, and we'll talk more about this one in just a moment, but there's also a basotracin disc on there. So we're covering 3 of the things that we need to be able to call this Streptococcus pyogenes, but we'll get to that more in just a moment. Now on this one, you can see again, the stabs are done right along the edge of quadrant 1. So again, on this plate, we have a STAB O positive, we have beta hemolysis. And we also have that basotracin disc there. So this is a plate that's been set up, and these are typically throat cultures. Set up a throat culture. This is what we'll do: streak it out, add a basotracin disc on it, and do some stabs. And that helps us to get to a quick result, quick turnaround time. So the basotracin disc that I've talked about a couple times now in those pictures, this is a disc that we will put down on the plate, as you saw. And this disc has the antimicrobial basotracin in it, and they stamp it with an A on top of this disc. Don't ask me why. I don't know why it wouldn't be a B or BAC or something like that, but it's got an A on top of it. And this would be recorded as basotracin susceptible or basotracin sus if we were looking at group A strep. So recorded as basotracin resistant, or res if it were to be resistant to it. But with Streptococcus pyogenes, group A strep, we would expect it to be susceptible. So what does susceptible mean? It would be observed as no growth around the disc and no zone of hemolysis. So that is what we would be looking for susceptibility. On this plate here, you can see clearly in quadrant 1 where this disc was dropped, whatever has been plated up on here, this beta hemolytic colony that seems to be pure is not hemolyzing around that basotracin disc. It's being inhibited, so it is susceptible to that. We also don't have growth up to the disc, so this would be basotracin susceptible. This is one that I would like to highlight because this is a typical throat culture that you would see. So we have clearly a beta hemolytic colony on here, and we have a streptolysin O or STAB O positive colony as well. So the STAB's positive, beta hemolysis. Now the basotracin disc looks kind of funny. So we have growth up to the disc, and there is hemolysis around, but it seems to be weaker around the basotracin disc. Now this would still be considered basotracin susceptible because even though we have growth going up to that colony, you got to think this is normal flora that we would expect to see in the mouth or in the throat. And so this isn't abnormal to have other things growing on there. We would expect to see other things growing unless the Strep pyogenes had just completely outcompeted everything and destroyed everything else in there, which would not be super likely. But we have growth up to the disc, and that's okay, but we can still see that it is inhibited and we have a lack of hemolysis around there. So this would be basotracin susceptible. Now, basotracin does have some specificity problems, some sensitivity and specificity problems. First off, 1% of the group A streps are resistant. So we expect them to always be susceptible, and that's how we really plan out the testing. But that's not always the case. 1% of these are resistant to the basotracin. And the second point is 5 to 10% of the streps are susceptible. So this isn't completely definitive in one way or the other. Basotracin by itself won't tell you if it is truly a group A strep. So a test that's commonly done in the laboratory is a throat culture, like I've already mentioned. And a throat culture is looking for one thing and one thing only, and that's group A strep. So we will call this a strep screen as well, group A strep screen. Now, essentially what this is just a plate that has been struck out, kind of like the ones we saw before, which are throat cultures. But what would happen is you would get a swab down. So the patient would look like they have these nasty white pustule type things on the back of their throat, or just a red throat, whatever. It doesn't matter. And even it's funny if physicians try and predict if it's strep or not, because they're wrong half the time, which statistically means they're pretty much wrong all the time. But not really. Anyway, so you have the swab of the back of the throat, gets sent down to the laboratory. You're sitting back there in microbiology processing, bacteriology processing. So you would then set up this plate. You'd take the swabs out. Usually they would do a dual swab, and usually that was just because you could do a rapid strep screen right there. So do a ELISA type test. And the second one was meant to streak out the culture plate. So you would streak out quadrant 1 with that swab, toss that swab, and then you would pull out your loop and you would streak out quadrants 2, 3, and 4. And then I would go back and take a little bit out of quadrant 1 and stab it in. So after you've done those stabs, you then drop a disc in the top of quadrant 1 and record that you have just set up primary plating media for a throat culture. You would incubate it. Next day you would come in and be looking for your results on that to say if it was either positive or negative. And it's a really quick test because we are just looking for one thing. And if the criteria are there for that one thing, we would call it positive. If it isn't there, we would call it negative. So to be able to report this out as a positive test, we need to have all 3 of these tests, these individual tests, come up positive. So group A strep positive would require good strong beta hemolysis first off. So if we have some good strong beta hemolytic colonies in there, that would be a positive. The second test on there, the streptolysin O-pos or STAB O positive, we would need that to also be clearly a strong STAB O. So we have that positive. And the last one there, the third, basotracin, I mentioned before that there is sensitivity and specificity problems with that. They're minimized, or they become very, very minimal when we compare that with the other 2 positive tests. So if we have basotracin susceptible, if we have STAB O positive and good strong beta hemolysis on that plate, then that's a positive group A strep or Streptococcus pyogenes throat screen. There are some within the throat that can give you a positive on 1 test or maybe 2 tests. And whatever those organisms are, they won't show up all 3. So even in combination, you won't get all 3. So if you do have all 3 of these come up positive, you could then report this test out. And you'd start with your plate rating. I'm sitting talking about this and not doing my duty of clicking the buttons. But if you had all three of these as positive, you could report it out, and in this case 3+ Streptococcus pyogenes or a 3+ group A strep. So again, you must have all three of these to report out a positive throat screen. So let's take a look at a bunch of these. So here we have Stabo positive. And you can see there's some other organisms there too. You would expect that, and they're growing right where you stabbed it into the auger. But underneath that, we do have a Stabo positive. We have beta hemolysis, and you can see the basotracin disc. It is susceptible. That disc is inhibiting one of those many organisms that's there. So we have good susceptibility basotracin to the basotracin disc, beta hemolysis, and a Stabo positive. Here's another throat screen. The disc is kind of dark up in the top left in quadrant 1, but you can see here again, do we have beta hemolysis? Yes, we do. Are the stabs positive? So Stabo positive? Yes, they are. And do we have susceptibility around that basotracin disc? Yes, we do. So those are all there. So this would be a 4+ group A strep. Another plate, this one is from our laboratory, and you can see some really good beta hemolysis on there. Just blow a hole through the plate, and those stabs, look at the stabs. You can see the writing on the other side of the plate very clearly, which is a 5 or an S, something like that. But then basotracin disc as well. Basotracin disc that's been dropped in quadrant 1. It is clearly inhibiting some of the organisms, because you can see there's not near as much growing up to that disc, and also the hemolysis has been stopped, completely stopped where that disc is. So this is a good example of a 2+, about a 2+ group A strep. Here's one that I would show some caution with. So we do have some basotracin susceptibility, it appears. And we have some beta hemolysis on there as well. These are lighter, kind of a white colony, and where I would show caution with this, though, is those stabs don't look positive. They're not any more hemolytic than around the colonies. So this one I would reject, or possibly work up further. Maybe take it off the plate to try and confirm it, just to double check it. But in just initial screening, this is a negative for group A strep. So on the culture report form, we will set this up, or whoever sets this up initially when it comes down, if it's processing. If it's you would want to record the date you set it up, and you'd write it as an SBA, basotracin, and stabs, so back and stabs. You'd come in the next day, so in this example it's been 10/3/17, 10/4/17. You come in, you look at the SBA plate, and you're looking for those three things. So you're just going to record those three things. You have 4+ beta, Stabo positive, basotracin susceptible. So you would then write out organism isolated would be 4+ Streptococcus pyogenes or 4+ group A strep. Now, if it's negative, you still need to report it out. So we don't just report it out as negative because it's not negative for growth. There's still growth in there. But what we will say is that it's 4+ normal flora, or we would say no group A strep isolated. So one of the two are good options, but we want to acknowledge we're not necessarily looking for anything else in there. So if we say the normal flora comment on there, that covers it and says that there is other stuff there, but otherwise you could say no group A. If that's all we're going to look for, then that's all we necessarily need to report. This plate here, we have good beta hemolysis in spots, Stabo isn't positive. We don't even have a basotracin disc on this one. So final culture results would be 1 plus normal flora or no group A isolated. So with that, we can add this group A strep screen, the back and stab plate over there, as another test that can be done to quickly get from initial setup straight down to group A strep. So this would be like taking a shortcut and not having to wait necessarily for growth to be able to test individual colonies. We just are looking broadly at this plate to see if we can call it out as a positive. Back to a few more images of this just to kind of pound this home. So this picture, what would you say? How about let's just start it off with beta hemolysis. Nope. Stabs, they're right there where they should be, right next to zone or quadrant 1. They're not positive. Basotracin disc is resistant, so no. Negative. No group A isolated. What do you think? Basotracin susceptible. Good, strong Stabo positive, and we have beta hemolysis. We've actually looked at this one already, but I want to bring it up again. Again, what do you think on this one? I'll give you a second. Alright, that was a second. I got arrows pointing to why you should say that it's normal flora. So 2 plus normal flora on this one. We have no Stabo positive. This is Stabo negative. We have some beta hemolysis in spots on this, but the ones that are looking beta hemolytic are large colonies anyway, large white colonies, so they don't even look like your typical streps. And no basotracin disc on here anyway, but not necessary because it's a negative anyway. Alright, try yourself on this one. Alright, so we have beta hemolysis. Those stabs are looking pretty fantastic there. Good, strong streptolysin O reaction. And we have susceptibility to the basotracin disc. We have a lack of hemolysis where everything else has been hemolyzed in that zone. And we have no growth right up to that disc. So this would be 4 plus group A strep positive. Alright, so just as a strep screen, there's a lot of the wonderful things that could be found in the back of the throat. None of them hemolytic, none of them looking like a strep, so this is 3 plus normal flora. This is a cost-effective way to set up multiple throat screens. Basotracin disc just dropped over the top because we still could be looking for those stabs, which are not present in any of these, right? Stab O, the streptolysin O, all negative. We have no susceptibility to these basotracin discs, and there is really no beta hemolysis on this. We have a darkening of the auger, but that is not what we're looking for. So with this, we wouldn't call any of these out as positive. I feel like you're probably getting this down pretty well, so let's make this the last one that we look at. What do you think? Of course, there is beta hemolysis, there is Stabo positive, there is susceptibility to the basotracin disc. Of course, this is positive. Group A strep pos. There's another plate that can be used for isolation of group A strep, and that is the SXT plate. And it's essentially an SBA plate with sulfamethoxazole and trimethoprim. So these two in combination will inhibit pretty much all of the normal flora that would be in a throat culture. And so you can plate it on this, it would knock down most of that. Now, some of the other streps can get through, but it even inhibits many of the streps. So really what you're left with is basically beta hemolytic strep. Now, it doesn't remove, and it doesn't differentiate between group A and group B. So group B could still be growing on this too. So you still need to confirm that it is group A with additional testing. That wraps it up for today. So we've talked about some of the primary plating of throat cultures and the identification of Streptococcus pyogenes. We're going to continue with some more identification of Streptococcus pyogenes in the next class, and some of the other methods like the PYR test, some of the commercial tests that are available, and we'll talk some more about, we'll talk a little bit about pathology, and might even get into some of the group B strep. So till next time. Welcome to class number 12. We are continuing with the lab identification of Streptococcus pyogenes, and after that we will cover the pathogenicity and the epidemiology of this organism. We've been talking a lot about the different tests, the bazotracin, the different types of hemolysis that we can see, and for the strong beta hemolysis when we've talked about group A strep thus far. So we're going to move down on this list and start talking about just PYR right now. So the PYR test, if we weren't doing an initial throat identification, if this came in some separate way, so we weren't necessarily just looking for group A strep, we may come across it still. So we may come across it in a blood culture or in some wounds or some other places. And when we do come across it, we need to be able to still identify it. So the next test on this list would be the PYR that we would do. But I have one quick note up here that I just want to say. Now, catalase test, PYR test, many of these spot tests, you need to make sure that you can see enough of the colonies on the stick or on the loop, whatever you're using to do the test. And so you want to be careful with that. PYR in particular, if you don't see enough of the colony and you can't actually see it when you smear it on the disc that we'll talk about in just a moment, then you may have a false negative. So same thing with catalase. If you're really not grabbing enough colony to be able to truly and confidently call that it's a positive or negative, then you're not grabbing enough and you need to get a little bit more. So I know you want to be sparing with some of it because you only had 3 isolated colonies, but if you don't have enough to work it up, then you need to sub it out for the next day. But that's my quick comment. So let's now look at the PYR. This is the PYR test, and it's a handy little test. It's really convenient, can be done fast, and helps a lot to differentiate certain organisms, one from the other. Now, PYR, this stands for pyrrolidinase peptidase, so PYR. And the concept of this test, and we'll go step by step through it in a minute, but we have this PYR disc, pyrrolidinyl beta naphthylamide, and this disc will break down the organism. So then we add the PYR reagent over the top of that, this cinnamaldehyde, and we'll get a reaction. But let's take a look at it in more detail on the next slide. Quick note before I move on, if you remember from the flow chart, this is there in conjunction with the bazotracin disc. So we could do the PYR or we could do the bazotracin disc. Remember, bazotracin disc did have specificity and sensitivity problems. And so doing that alone isn't as ideal, but the other problem with that is that's an overnight test. When we're looking at the PYR disc, this is very short. This is a few minutes that it would take to get it done. So the PYR disc is considered a spot test, and we quickly have a result. So then we could then send it out, result it out, final it out, and be done with it. Or we would then know what else we need to set up instead of waiting another 18 to 24 hours to be able to figure that out. So that's an important part of this. But the PYR disc, this is based on a couple things. So we have this disc, the pyrrolidinyl beta naphthylamide. And if you add the colonies on, so initially we'll take a little bit of saline or some water and we'll damp that disc. And when you do this, after you'll take I don't know, 3 to 4 colonies or so, and you'll just swipe those on the disc. And you need to let it then sit for about 2 minutes. So what this is checking for is if there is that PYR enzyme there. And if that enzyme is there, then what it's going to do is it's going to break down the pyrrolidinyl beta naphthylamide into L-pyrrolidone and beta naphthylamine. And the beta naphthylamine is the important part here. This is what we're looking for, and this is what would react with our reagent. So after 2 minutes, After we've had it sitting on there and we've allowed it to break down and a lot of this beta-naphthylamine to be created, if it does truly exist there, we'll then add the cinnamaldehyde over the top. Now the cinnamaldehyde's a little reagent that they have in a dropper bottle. So you'll add that over the top, put a drop on it, look for development of a red color. If you get a red color within about 2 minutes, typically 2 minutes. There's some that may take a little bit longer to develop, but 2 minutes is generally as long as you need to wait to be able to look. You see the development of that red color, and this would be a positive result. This is what it would look like when we have a result. So on the left we have a PYR positive. So this has that beta-naphthylamine that has reacted with the cinnamaldehyde, and that's produced this red color. So we do have that PYR enzyme in there. And on the right is a PYR negative. Now this is a very negative PYR. Sometimes, and I want you to be cautious about this, sometimes you can get some color. You can get a peach color or something like that. We're not calling peach positive. You need a good red color to be able to call this one positive. Because sometimes it can just be the colony, sometimes you can have some weird reactions with it, but it goes red if it is a true PYR positive. So be careful with that. Now my other comment from this slide is this isn't done in conjunction with the throat screen. The throat screens are themselves totally positive or totally negative. So that's a test in and of itself, and it's made to 100% identify group A streps. So we don't need to be doing any additional testing on that. If we have all of those three criteria, if we have STAB-O positive, if we have basotracer susceptibility, and we have a strong beta hemolysis, then we have everything we need to call out a group A strep. We don't need to be doing anything else. Now, this is, like I said before, if we're just going down that flow chart. So if we're trying to move down it, we have a beta hemolytic colony on an SBA, it's catalase negative, gram-positive cocci, chaining, not chaining. We would then move to this test. So we would do the PYR test, and this in itself is 100% definitive for group A strep. So if all of those other things are there that have made us move towards this with the flow chart, a positive PYR result would positively identify group A strep, 100%. So again, do not use this to confirm group A strep throat screens. It's not necessary. It's a waste of time, waste of money. These are kind of expensive tests to actually run, these spot PYR. The one other thing I want to talk about with this is what you need to know about this, because these are some super long names. I'm not going to require you to do a fill in the blank or anything like that. Like, you're not going to have to be able to spell this all out. But I do want you to recognize these names when you see them. So if I give you a list of different reagents and say, what would you need to do a PYR test, or what's involved in a PYR test, I would expect you to be able to identify those and be able to pick out the names that we're using. So that's what you'll need to know of the PYR test. Again, it's a very useful test and can save you a lot of time, can greatly decrease your turnaround times. One last precaution, and I've already kind of warned you of this precaution, but I'm going to hit it again. So in the testing we're going to be talking about coming up, and in the PYR, and again the catalase, you need to make sure you have enough organism. So not enough organism on the PYR disc. If you don't get enough on there, you're not going to get enough breaking down to see a good color reaction. And what you're probably going to do is you're going to see a faint color reaction, and it's just going to implant doubt in your mind. And so you're going to end up repeating the test again, and you're going to use up more colonies that you didn't have to use in the first place. So you want to get enough colonies on there to be able to truly call it a positive, and you don't want to run across any false negatives by having too few colonies that you think it's a negative. There's a couple more tests that we have to talk about on this page still, and those are the rapid group A strep tests, and there's also the latex agglutination test. So we're going to talk about the rapid group A strep tests right now, and spend a little bit of time going over the procedure for that. Now, this is an example of one type of rapid group A strep test. And these are commonly found throughout any clinic or doctor's office. These are low complexity. So according to CLIA, you don't have to have a lab degree to be able to run one of these. They're often put off in a stat lab or something that's just right nearby that usually has a phlebotomist that can run them, or an MA. And these can be an initial screening thing. So they're quick. If somebody sees something that looks like strep throat, they can do a swab, go in, run it, and then confirm it with the lab later if it comes up negative. But this is just one example, and I have one more on the next slide. So this is the one we use on campus, and this is Signify Strep A kit. And you can see here this cassette on the left, we have a window that has a C, which is the control line, and a T, which is the test line. And then we have a well off to the side of it. So what we would be doing is extracting, taking whatever that swab is that came down. Usually, like I told you before in a prior lecture, you might get 2 swabs. You should be getting 2 swabs to do this. And so the first swab would be used for this test, for the rapid strep test. You would put it into a thin little plastic tube, try and extract it with some extraction reagent, which would then pull the antigen or that C carb out of the cell wall. And then we would, once we've let it sit for a minute, put some drops of that into this well on the right hand side of the cassette. And we'll do that with a wash solution, and that will help it move across. And if group A strep in this case, because that's what we're looking for, if that is present, it'll show up as a red line on the test line. Or if it's not there, we'll just get a control line. That's all we'll see is the control line to let us know that the test worked, but there was no strep there. All right, so what is actually happening? We're going to go over the details of this. So, throat swab. Patient comes in, sitting on the physician table, their throat sore. Physician looks at it and says, yeah, it looks like could be strep throat. Let's test for it, or let's at least screen it out. So they take a couple swabs, go back to the back of the throat, make sure they swab back of the throat really well, and then send it down to the laboratory. Laboratory will then take one of those swabs, and after all the computer work's been done, it's been punched in, you got your labels, all that stuff, they'll then pull out the test kit, the rapid strep test kit, add a couple of drops of an extraction reagent to a tube, to these plastic kind of flexible tubes most of the time, and then you'll take the swab and you'll put it in, and you'll kind of ring it around, smack it around, try and get it all of the potential strep to react with that extraction reagent, because what it's doing is pulling that group A strep C carbohydrate out of the cell wall. So we'll get that out of it, and then if it is there, we'll pull it out. The next step would be to put it onto the filter paper. So the well that we have there, we're then going to wring this swab out, and you want to make sure you do this. I like to get all the juice off the swab, and since it is a flexible wall tube, you can kind of pinch it and make sure that you get all the juice off. So you'll then add it into that well. And in the well, this is acting like a filter and it's moving through. But at the same time, we're going to add this antibody enzyme conjugation. So conjugated is just a fancy way of saying combined. So an antibody towards group A strep has been combined with an enzyme. So these two are stuck together. Now, this is specific to group A. So we have group B down here. It's not going to react with it because it's that whole lock and key concept. So specific to group A. If group A exists in there, the conjugated enzyme antibody will attach to it. So this is just hanging out on there. And then we can add the substrate over the top, which will then move in and react with the enzyme. When it reacts with this enzyme, we get the color change, and that's what you see. Now this test is done all the time in the clinical lab. It's an ELISA test. It is enzyme-linked immunosorbent assay, and done all the time. We do these tests a lot, so you really need to commit it to memory. An ELISA test, we can really use this for a number of different organisms too, because if you think about it, if we're testing for a specific antibody that we make towards an organism, we can find a lot of these to do to add an antibody against. And when we do this, we can add a conjugated enzyme to it, and we can test for about anything. So really useful test, need to be able to remember it. It's something that comes up again and again. I'll test on it, and the BOC will test on it, so you need to know it. Let's look at that cassette again, and I actually want you to draw this maybe at the bottom of 2-18 where it's talking about the ELISA method, because I want you to understand this concept. Now, when these are manufactured, they come with antibodies stamped onto them. So IgG that is specific to the group A C carbohydrate, and these are fixed to that filter paper. Now, we'll come in and we'll add the patient C carbohydrate extraction. So we should have that group A C carb extracted now. And we're going to add it into this well. And when it does it, it's going to flow along. So it's going to move laterally across this thing. And if it runs into that antibody that's been stamped on there that's specific to group A, it'll get stuck. It'll stop there. And on the other hand, if it isn't there, everything will just wick along and nothing will get stuck there. Now, at this point, we would add that antibody that's been conjugated with enzyme. So we have really two antibodies in this case, two of them. So one of them that's been fixed to the paper in the manufacturing of this, and another one that we're going to add. So we add this antibody conjugated with the enzyme, and it's going to move across. And if it now sees this group A C carbohydrate that's been stuck at that test line on there, or on the control line, it's going to then attach to it. It'll attach onto that. So then that final step, we'll add the substrate across there too. So when we add the substrate in, it's going to wick across, and if it finds that enzyme, the antibody conjugated with the enzyme, it will attach to it, and that's what will give us this color reaction. Here is an ELISA test, but produced by a different manufacturer. This is produced by Abbott. And this one, maybe you're familiar with, or, well, it works on the exactly same principle. We have a hole in the bottom of the card on the left, and we're going to add the reagents and the extraction C carbohydrate. It'll wick through. We have the second window, which will produce the positive or negative for the organism that we're testing. And then there's a control bar that will light up if it's working properly. I have a couple more comments that I want to make on page 2-17. Now, the Rapid Group A Strep Test Kit, remember, this is part of this whole double swab thing. So it comes to the laboratory, the first thing they want to do with those double swabs is to do the rapid strep. So double swabbed in the back of the throat, we get a little bit of both on there, comes down, we take one off, rapid strep kit with that. Now, if that's negative, we will do an SBA back and stabs plate. So we're going to be testing still to see if we can find, if we can still find strep pyogenes in there. And why would we do that? Simple. The kit is only 90% sensitive, and it has improved slightly over the years. It's just not reliable enough. And so we do still worry. Strep is a very dangerous organism. Very dangerous organism. And so if there is 10% of those out there that are being missed, and these people could potentially be walking around still with streptococcus, then we want to know about this. So this is especially important on those that are under 18, because we know that the majority of the bacterial infections, or the majority of the sore throats, are bacterial infections in younger kids. And so with that being said, there's a higher proportion and there's a higher danger associated with those under 18 still having it. So where this kit is only 90% effective, the negative rapid test kit results need to be followed up by culture. And like I said, especially on those under 18, especially on minors, young kids. So may not be that way on adults, but it is dangerous enough that we want to watch that and follow it up with culture if we suspect that it is there. When it comes to reporting out the results, we're going to report them out either as positive or negative. Group A strep pos or group A strep negative. We're not going to do anything like normal flora. We're not going to make any comment about that. We're testing to see if the strep is in there. So simple enough, group A strep pos or group A strep negative, this is what would be reported to the physician. On to the last test that we will do for Streptococcus pyogenes, and really this isn't just pyogenes, this is all of the Streptococcus, the different species, almost all of them. Now, the latex agglutination kit exists, and this will test for group A through D, F, and G. So the way it's doing this is it's actually testing for that C carbohydrate or the lipoteichoic acid. And so we go through an extraction step in this, which makes it a little bit more tedious than just doing the staph test, the Staphylococcus aureus, the rapid test for that. Because this one we have to add in the organism into an extraction liquid, extract it out, let that sit for a minute, and then we have to add that over onto the card. So it's an extra step, not super complicated though. And then you can see all of these different tubes up there, or dropper bottles, have A, B, C, D, F, G written on them. And you would add this to the side of the card, and then you would add your sample second, and you always want to make sure that you add the reagent first and then add your sample so that you're not possibly cross-contaminating these dropper bottles. And then you would just mix them together and then roll it around for a couple minutes and see if you see an agglutination reaction happen. So on this slide, I have these notes all written down so you don't have to jot these down, but I just want to highlight the point here that we aren't able to test for the non-Lancefield type with this. We're only testing for the C carbohydrates and the lipoteichoic acid. So if we're able to get these ones, then that's great, but the other ones would have to be tested in different ways. All right, with that, let's jump into now the pathogenicity and epidemiology of group A strep. A few slides back I mentioned how dangerous group A strep can be. And people know it typically as an inconvenience. They know that it causes a super sore throat. Feels like you've been kicked in the throat by a combat boot. So people know it in that way, but it is far more dangerous than that even. So we're going to go over the pathogenicity, and we're going to go into the details on each one of these and what makes it so dangerous. The first virulence factor that we're going to talk about is the M protein, and this is a major protein that exists. It's present in all strep pyogenes, so present in all of them, doesn't fall off anywhere. And it's also unique to Streptococcus pyogenes. We don't see it in group B or group C or any of those others. It's only in group A. Now, this is also attached in the peptidoglycan, attached in there, and it makes it accessible. And the accessibility of it is important just because this is what triggers the serological group reaction. So there's 80 different strains of M protein, and they're labeled M1, M5, M15, M75. So 80 different serological groups of this. And this is the reason why you can get strep throat multiple times, because if you were to get a M15, so you get strep throat, M15, your body's going to make an antibody to that M15, and this will give you lifetime immunity to that specific strain of Streptococcus pyogenes. But now later on the next year or even the next week, perhaps you come down with strep throat again if you were that unfortunate. And in that case, it would be a different strain. It would now be M64. And so this is a reason also why it makes it difficult to immunize against it, because there are so many different serological reactions that can happen with this. So 80 different strains, and we have to, have to be aware that that's why that happened. So I guess you could be totally immune to it too if you had been sick with 80 different strains over your lifetime, but that is pure misery. I don't know. I wouldn't wish that upon anybody. But functions of this: it will resist phagocytosis, and the second point is that it adheres to mucosal cells. So you're out there breathing things in, somebody coughs in your face and you inhale instead of holding your breath like you should when people cough in your face. And this will help it get in there. It'll attach that mucosal lining in there, and then that's how it's able to initiate infection. Now, one other point I want to make about this M protein is that this is very similar to the Staphylococcus aureus protein that we have in there. Acts a lot the same, resists phagocytosis, and helps in that same function that Staphylococcus aureus, the protein A, does. So just wanted to make that note. The next virulence factor that we're going to cover is DNase, and this is a useful one. You have that ACE on the end, so DNase. ACE meaning that it's an enzyme. So this is an enzyme that will break down DNA. And its function in there is really to give the bacteria a little bit more room to move. So this is a pyogenic organism, so it'll create pus as we have dead and dying tissue and the cells die, the bacteria die off. So if you've ever done a DNA extraction, when you do the extraction and you break open these cells like what's happening here, it becomes a lot more viscous and so more difficult to move around in. And what this DNase will do is it'll come in and break that stuff down and give it a little bit more room. I don't know, it's a weird thought, but I always think of this as like a snowblower in that heavy snowstorm. And because I can think of pus as the white snow there, and I just think of the snowblower cleaning it up and getting it out of the way type thing, chopping it up and moving it out. So that's really kind of the function of the DNase, though, is to allow it to spread easier into other tissues because it doesn't have to move through such a viscous fluid. The next one on the list is hyaluronidase, and this one, we've actually learned about this one before when we learned about Staphylococcus aureus. Now, hyaluronidase is also known as spreading factor, right? And this will go through and break down the connective tissue. It breaks down that hyaluronic acid that exists in the connective tissue, and so this helps it to spread around a bit easier. The streptokinase, again, this is another enzyme. This is a kinase much like the staphylokinase that we saw before. And same purpose as before. This is used to break up clots, so it will destroy that fibrin clot that exists, and it helps it to spread and move a little bit easier. I showed you this image before, the coagulase and kinase, and this is a kinase, so same idea. It'll come in, it can help break up the clots and release bacteria. And now we have streptolysin O and S, and we've talked about these when we're looking at them on an SBA plate, but these are virulence factors that again could be used in a person, so in vivo. And in a person, this is another useful thing. So much like on the plate, they're able to break down red blood cells in the presence of oxygen or the absence of oxygen. So again, another useful virulence factor. The last one here is pyrogenic exotoxins. Now, this is a group of 3 distinct toxins, so completely different toxins that will cause macrophages to release cytokines. When it does this, it triggers fever, rash, and shock. So this can be very similar to the toxic shock syndrome that we saw before. In fact, it is a toxic shock syndrome that's associated with group A strep. And we'll talk about these toxins in more detail when we get into the disease states that exist with these. Scarlet fever is one of those as well. But again, these toxins, they cause the macrophages to release their cytokines, which then causes all sorts of problems. Now we're going to be talking about not just the virulence factors, but the diseases of Group A strep. And in this section, I've categorized it based on localized infections and sequela diseases. Now these are the ways that we parse it out. Two separate types of infections that you can get with it. Now sequela will be ones that come on after the fact more so. And the localized infections are the rapid onset type of infections that we'll see. And the localized infections are typically suppurative and purulent. So this is pus producing, right? We see an increase in pus or fluid or a buildup of some sort that comes fairly rapidly. The first one that we're going to cover is pharyngitis. Now pharyngitis, you all know this one. This is strep throat. This is when we talk about Streptococcus pyogenes, we typically talk about strep throat. And the symptoms of this are an abrupt sore throat, horrible sore throat usually, and you've had it. I've had varying degrees of these sore throats. I don't know, 7-8 years ago was infected with influenza and strep at the same time, and felt pretty crappy. Throat wasn't so bad though at the time, so that's why I didn't think it was as serious. I didn't think I had strep throat actually, but it was a mild sore throat. So you can get a varying degree of that sore throat, but then sometimes, like I mentioned before, it feels like a combat boot to the throat or a blast to the back of your throat, and every time you swallow it's just cringeworthy. And I feel like I just made you feel that because you've had it before and you know it. So you swallow and it's just painful. You don't even want to swallow. It's not worth it. And so the abrupt sore throat, then you have malaise. And malaise is just that overall rundown feeling. You just lack energy, you feel like junk, you just want to sit around watching Maury Povich, not really eating anything because, again, it would, feel like a cheese grater on the back of your throat. But fever comes with this and headache. So if we're so inclined to look into somebody else's mouth who says they have a horrible sore throat, you might see this. Or if you just want to look in your own mouth and see if you can see any of these. If you have a sore throat, then oftentimes you will see some pustules, or you'll see some red spots, or you'll see something that's happening back there, because that is where the pain is coming in. You have this dead and dying tissue in the back of your throat. It is just destroying the tissue back there. So this one on the right is a crazy example of it. You can see just how big those pus pockets are in there. And it is stereotypical. You can often see these pus pockets, and this is where you'll get physicians predicting, like, yes, I know this is Group A strep because I saw those pus pockets back there. They're not always correct in that, but that is very, very stereotypical for Group A strep. Now, pharyngitis, I have a couple more notes on this, and the first one is that it's most prevalent ages 5 to 15. And when it shows up this way, it's 90% bacterial. Of the bacteria, 90% of those are going to be Group A strep, and 10% of those are going to be viral. Now, I want you to think about this, and I want you to think back to what I was saying earlier about how a rapid Group A strep, especially in those that are under the age of 18, should always be backed up by a throat culture. This is why 90% of the time it's going to be bacterial, 90% of that time it's going to be Group A strep. So there is a high percentage that this is Group A strep in the younger people. So where it is so dangerous, and we saw the virulence factors, and we're going to talk more about other disease states as well, we need to make sure that we're catching it. So we don't want that 10% of the patients to walk out of the clinic thinking that they're okay, when all it would take is a simple antibiotic to be able to kill it. Now, look at the minors in contrast to adults. Adults, we have 10% of those bacterial infections and 90% viral. So this is the reason that as you turn 18 and all of a sudden it flips that percentage, which is not totally true. But once you get a little bit older, it becomes less important to do that backup screen because percentage-wise, it is a much lower percentage. Now, some facilities choose to back up every single rapid strep with a culture. Not always necessary, and so others have chosen to go away with that. And the last point is something I've already mentioned, is that M protein antibody. So, like I said, you have lifetime immunity to that one that you get. So, this is a reason that it somewhat flips for adults because they do have immunity to some of these common strains that go around the public. So, you're not going to be getting as often the sore throats as the younger kids or getting the ones that are bacterial because you do have some immunity against that. But you would be more susceptible to the viral because viral, you don't often get immunity or lasting immunity to those. The next localized infection we'll talk about is pustule impetigo. And pustule impetigo is caused most of the time by group A streptococcus. And remember, group A streptococcus can cause another type of impetigo. Do you remember what that one is? Bolus can cause bolus impetigo 20% of the time. So, impetigo is a pustule caused by group A strep. Now, it can also cause pyoderma. You can see the text, page 545, for more information about that, but this is a confined pus-producing lesion. Erysipelas, this is a strep infection of the skin that involves the surrounding lymph nodes, and you have clear demarcations on there. It occurs most commonly on the faces of children. And cellulitis, this may develop, and it's just a deeper invasion with group A strep, so it gets into the deeper tissues. May be serious, can even be life-threatening, where the bacteria have the opportunity to go septic. So cellulitis can be very dangerous. This is a photo of a man with erysipelas, and you can see, like we just defined, these clear edges on there. So you can see the line and the redness that pretty much stops at a point there. It doesn't spread out. It's, I don't know, the localized tissue right there, all involved. And again, this is erysipelas, and this is caused by group A strep in this case. Can be caused by other things, but this is a group A strep infection. This is a photo of a former student's son, and this is a classic example of erysipelas. And you can see the clear demarcations on there, and she had mentioned that it was very itchy, so he'd just itch it and spread it around, and it would just spread quickly through the tissue. So yeah, good example of erysipelas caused by group A strep. The second major group that we're going to go over is the sequela diseases. Now, sequela diseases are also known as post-streptococcal diseases. So sequela diseases, post-streptococcal diseases. Same thing. Now, sequela diseases, if you can see in there, it's got the word sequel. Now, kind of like a sequel of a movie. Hollywood likes to constantly do more and more sequels of something that's been successful. So, kind of a similar thing. This is the root word of sequel actually means to come one after the other. So, sequela, this is one thing that's going to come after another. So, this is the next thing after that initial strep infection. So, sequel, and kind of like the movies, sequels, yeah, lots of, if we're talking like Fast and Furious, holy sequels. I think they're on Fast and Furious 10 now or something like that. You can even have your main actor pass away and just moves on, because I guess Fast and Furious is it's a thought. It's a religion. It's not one man. It's not ended by your main actor passing away. But anyway, same thing. Lots of other sequels to come for strep pyogenes, or potential sequels. The first of the sequelae that we're going to talk about is scarlet fever, aka scarlatina, aka, just kidding, there's no more, just scarlatina. So scarlet fever, and this will appear after 1 to 2 days of the pharyngitis. Now this is Streptococcus pyogenes that is releasing those pyrogenic exotoxins. So we talked about those pyrogenic exotoxins before and the way it can induce fever and some of these other things. Now when it does this, it produces a rash, and this rash will typically begin on the chest. And the rash will go away after about a week, so it can last a little bit of time. Now it will spread quickly across the body, and it'll cause a sloughing off of the skin. Now when it spreads across the body as well, it will accumulate. So the toxin really emphasizes it in the tongue. So it attacks the tongue, and you'll get this strawberry red tongue, this bright red tongue. And it's really one of the hallmarks of scarlatina or scarlet fever. I know color can somewhat get washed out in these images, but this is a bright red tongue. So this would be that strawberry colored tongue that we attribute with that pyrogenic exotoxin. So the next on the list is rheumatic fever. And what this is a attack really on the heart valves and the muscle. So this inflammation and the bacteria getting in there will cause an immune response, really. So this immune response causes inflammation. This inflammation damages the heart valves and the muscles surrounding the heart. So that immune response, we have antibodies that are targeting the bacteria. They cross-react, and it's thought to be that they're very similar with the heart antigen. So they get confused, they attack it, inflammation and damage overall to the heart and the heart valves. Which again, you lose the heart valves, you're in big trouble. So this is common mostly in 5 to 15-year-olds, which is our same range for developing strep throat. But even though it's common in that age group, it's fairly rare in the United States and other developed countries. We mostly see rheumatic fever in some of the developing nations. You may have studied before about glomerulonephritis, and probably in urinalysis. Acute glomerulonephritis is, in this case, where we have the antibody and antigen compounds not coming out of the blood. So they're not getting removed, they're not getting removed by macrophages, they're just stuck in there. So as being stuck, we have our filters of the blood that come through, and they get stuck in the glomeruli of the nephrons. So these are tiny little systems that are meant to filter through the blood and separate out for urine purposes, right? And so they get stuck in these nephrons, and when they do this, it causes an inflammation of the nephrons, and you get reduced blood flow, and then that just continues and continues, and you have less and less blood flow. What happens in this case is you'll get hypertension and low urine output. Really bad thing, can lead to destruction of the kidneys. The next on the list is invasive group A streptococcal disease. And this is also known as AKA necrotizing fasciitis. And you may have heard the term before, necrotizing fasciitis, because this is an attention getter. This is one that shows up in the news. So you put necrotizing fasciitis on the title and people click on it. And it's the same thing as zombies. And yeah, don't get me started on zombies in the news. Zombies are the most irritating thing to me. And I'm sure I've upset a lot of you who are avid preppers in the zombie apocalypse. But anyway, so necrotizing fasciitis. What's happening here is group A strep is going to secrete a bunch of enzymes and toxins that are going to destroy the fascia and the connective tissue around the muscle. When they are destroying this, they are moving quickly, just site to site, and organism has been reported to move about an inch an hour. An inch an hour. That's insane. Wild to think about. Terrifying. Could see why it would make the cover of newspapers. But with it moving so fast, the bad part about that is amputation is sometimes the only way to stop it. And even in doing this, 50% of patients will die from this. So it is a terrifying sequela disease that's attributed to group A strep. Now, one more point on here before I move on. I said it secretes enzymes and toxins that destroy the fascia. So what factors would lead to the organism spreading so quickly? Just want you to think about that for a second. We talked about them. This is all the virulence factors we've talked about, and it's pretty much all of them in combination. So to be a necrotizing fasciitis, you have to have a lot of these in there. So DNase, it would need to be able to break down any of the dead and dying tissue to be able to move and spread easier. Streptokinase, be able to break up those clots that could form. Hyaluronidase, this one's very important for it to be able to spread so quickly because it needs to be able to break down that hyaluronic acid in the connective tissue around the muscle. Exotoxins, that would trigger immune response stuff, and you would just have that inflammation that attributes to it. And so all of these things will result in massive tissue damage and rapid tissue damage. Gross image alert. This one's pretty upsetting. This is somebody that had necrotizing fasciitis and they've just cut the skin out. Cut it out. And it looks like they've stuffed a little piece of tissue in there. And I am not 100% on the treatment, but this may be soaked with antimicrobial. That's just a guess. I don't know, or it's just sopping up some of the fluid. I have no idea, honestly. But this is disgusting. This is one of the ways that you have to treat it, though. Horrible to think about somebody just cutting your skin out or having to amputate your leg, but this is the reality when you have a disease like this. The last one we're going to cover is streptococcal toxic shock syndrome, so STSS. This can spread from the original site of infection and get into the bloodstream where it would cause septicemia. Now, if it gets in there and causes septicemia, septicemia is where it's growing and causing symptoms, right? We typically only see this in immunocompromised individuals. But if it does spread from that original site of infection and it starts replicating and causing symptoms, the symptoms we may see are inflammation, pain, fever, chills, nausea, vomiting. All of the toxins and everything else that Streptococcus pyogenes is capable of doing will lead to shock, organ failure, and death. Over 40% of patients will die who become septic with Streptococcus pyogenes. Very serious and just one of the many sequela diseases and the dangers of having group A strep and not getting it treated, because these things can form a lot easier. So this is the importance of going in and getting checked. If you do have a sore throat, making sure that it's not strep throat. This is why physicians are so crazy about it too. Like anytime you even say anything about a sore throat, it doesn't matter if you're in the midst of flu season or anything, they'll always test for strep throat because they want to rule this out, because there are so many dangerous other sequela diseases that can be attributed to group A strep. This is Jim Henson, and he's really kind of like the god of the Muppets. He was the creator, and he had his hand in everything Muppets. So Sesame Street, the Muppets, everything. He was involved in it. Kermit the Frog, clearly one of his most famous creations, and Kermit's girlfriend, Miss Piggy, had an important part in that too. But Jim Henson died in 1990, and it was initially reported that he had strep pneumoniae, but it was later determined to be streptococcal toxic shock syndrome that he died of. So again, a sequela disease of group A strep, and I don't know, this photo is pretty heart-wrenching to me. So sad. Kermit just remembering his dad there. So anyway, something to be careful about. I want to just reiterate this point, that the sequela diseases show the importance of diagnosing pharyngitis that is caused by group A strep. With any sore throat, it's important to test for group A strep, and if we have a negative rapid strep kit result, then we need to follow that up by culture. Now, I say these things, and something that's becoming more and more prevalent is some rapid identification using DNA-based tests, and that's great. That's happening in some of the bigger laboratories, and I'm sure that cost is going to get down enough that it can be done in smaller laboratories. But for now, we still have that rapid strep kit test that's being done in many doctors' offices and clinics around the country, around the world. And so they aren't perfect. We need to follow those up with culture. The last point that I want to make about Streptococcus pyogenes is the antimicrobial therapy that we would use for it. So antimicrobial therapy for infections with Streptococcus pyogenes would be penicillin. For group A strep and also group B strep, we call them the beta hemolytic streps, penicillin is very effective. These two organisms have not developed any type of resistance to the penicillin like we saw with Staph aureus. So we would use probably erythromycin or cephalosporin to treat this. And back to Jim Henson. So I'll leave you with this picture. I'll leave you on this somber note. After Jim Henson's sudden death on May 16, 1990, people across the world remembered the Muppets creator with tributes and honors. One of the most touching illustrations was this one done by Disney artists Joe Lanzicero and Tim Kirk. And in this tribute to Henson, they have Mickey Mouse putting his arm around Kermit the Frog, consoling him, because clearly he's upset with the loss of Jim Henson. So this appeared in a couple Walt Disney's magazines around that time, but I think it's just a sad image, and it just showed how much everybody was affected, especially in this realm, because he was such a giant in the Muppet world. So kind of interesting, but that concludes this class, and next time we'll pick up with Group B strep.